Good afternoon, ladies and gentlemen, and welcome back to the Global Young Scientist Summit. The first speaker for this afternoon is Sir Tim Hunt. His talk is entitled Lessons from a Life in Science, How I Stumbled on the Secret of Cell Division. The Q&A session will be moderated by Prof. Ng Shu Yen. Sir Tim, please.
I'm not allowed to walk around, which is very inhibitory, particularly as I actually can't quite see the slides from where I'm standing. And I must say, this is really quite an intimidating experience for me, because I haven't given a talk for, well, since 2019, I think. And I was used to giving, lots of talks. And it turns out that if you don't give talks, it makes you very nervous. So the first lesson is give lots of talks. It really helps get over those stage fright things. Second problem, I couldn't find, and the last 6 years we spent in Okinawa, and we just moved back from Okinawa to London. And in that move, my green laser pointer somehow got mislaid. So all I have now is this wimpy little thing, which I barely, can you see it? It is there, actually, but it's weeny. You want something much beefier to thing. And finally, compared to the previous speakers, I really am a terrible old dinosaur. The work I'm going to describe to you was done a very long time ago. But it's the story of my life. And I think there are some lessons to be learned there. And I hope you enjoy it. And I apologize for not being up to the minute. Actually, I stopped being a scientist. I stopped running a lab in 2010. And since then, I've sort of, it's kind of interesting. I always thought that Max Perutz, after he solved the structure of hemoglobin, had done something so miraculous and wonderful that why did he go on? He went on until he dropped dead, more or less. On the other hand, Fred Sanger double Nobel Prize winner, my wife happened to sit next to him on the plane going out to Stockholm. And they fell into conversation, and he said, "Oh, I was so glad when I could retire," he said, "because it was so hard." Interesting. And everyone was amazed because when Fred turned 67, which was the retiring age then, you know, suddenly, the day before he'd been in the lab, the day afterwards, he was nowhere to be seen, and the lab was completely clear. And everyone thought that was amazing. But he'd done so much, you know, and as he said, it was so hard. And I think, don't kid yourselves, it is hard. It really is hard. My thesis advisor told me, you know, "Tim, this business it's a manic depressive business, and it's mostly depression." As the previous speaker said, you know, I mean, mostly your experiments don't work, and most of your time, in my line of country, is figuring out why. And good people are good at solving problems and finding out what went wrong. So my first problem was to find a project, and the first project I chose, perhaps luckily for me, didn't work. And after 6 months of my PhD, I had absolutely nothing to show for it whatsoever. Turns out I hadn't read the materials and methods of the paper I was following carefully enough, and I was using the wrong strain of rat. So then I went to my first scientific meeting, and that was what really set me on the path almost for the rest of my career. Yes, for the rest of my career, really. There were 2 talks. First was an introductory talk by the guy on the left, Henry Borsuk, who talked about 2 totally unrelated things. One was the control of protein synthesis in red blood cells, and the other was the control of protein synthesis in sea urchin eggs. And the 2 really have nothing in common. A red cell, after all, is dying, it's lost its nucleus, and it just makes a lot of hemoglobin in the last few days of its existence, whereas a sea urchin egg, of course, is going to divide and turn into a baby sea urchin. So the 2 sides of the thing. But so the problem, the talk that really got me started in research was by Vernon Ingram on the right. He was the man who solved the chemical basis of sickle cell anemia. And he was interested in the question of the coordination of heme and globin synthesis. And so here, for those of you who don't know about hemoglobin, this is not actually hemoglobin, it's myoglobin, a closely related one, but the heme is the red thing in the middle, and it's surrounded by the protein. You can see what protein crystallography was like in those days. This is known as the sausage model of hemoglobin for obvious reasons. And you can see that they didn't quite have the connectivity of the chains in those days. Why is it doing this to me? Don't. Boy, oh boy. This, I should say, one of the other things I don't know, I've never given a talk with this computer before. It's sort of top of the line and everything, but I obviously haven't got it quite set up right. Never mind. So the question was, when does the heme get in? Oh, no. Now the thing is not responding at all. Something is really not right about this. I don't know. Hey, this is what happens when you haven't given a talk on a computer for ages. Ah, great. Why did that bring it back to life? I don't know. Okay, so as you know, proteins are made with messenger RNA, and the question was the heme goes in about 2/3 of the way down. And what Vernon said was that the reason why, if you didn't have any heme, you didn't make any protein, was because if the growing chain didn't get the heme inserted, it stopped, it got held up en route. And he presented evidence that that was the case. And I thought that was amazing. Went back to the lab and told my mates, and somebody who was a lot cleverer than I was pointed out that actually his data, if anything, showed exactly the reverse. He had completely misinterpreted his data. So we decided that we'd see for ourselves. You know, was it true that when you didn't have any iron around or heme around, that the ribosomes formed a queue. And we worked out a slightly better way to do the experiments, and we did the experiments. And when I say we, I was very lucky. The guy on the right, Lou Reichhardt, had worked, he came for a year and occupied the same bench as me, and had worked on rabbit reticulocytes in Harvard and learned how to do them. And Tony Hunter on the left joined the lab a year after me, and basically Tony and I collaborated on our PhD and looked into this business. And then I went to another scientific meeting. This would have been a year later, or even 2 years later maybe. And I met Irving London, and he was interested in exactly the same thing. And I eventually joined his lab as a postdoc. And by then we could make cell-free systems from these reticulocytes. That had been invented up the road, unbeknownst to us. Lou was actually interested in the coordination of hemoglobin synthesis. Could never get the cell-free system to work. But it turned out to be terribly easy. I mean, unbelievably easy. You make rabbits anemic. You then harvest the red cells, wash them, and simply add water. And if you add an equal amount of water, the cells burst by osmosis, because they have lots of aquaporins in the cell membrane, and you get a cell-free system which will perform exactly like the cell performed, even though its contents have been diluted in half. Of course, you have to make the salt concentrations right. You have to add an ATP generating system and various other little tricks like that. And as you can see from the graph on the right, if you leave out the heme, what happens is protein synthesis starts off at the normal rate. And it really is exactly the same rate as it would have been in the intact cell, to work out how fast they're moving. And if you add heme, it carries on at that whole rate for more or less as long as you have the patients to conduct the experiment. And moreover, if you delay adding the heme and then add it back, protein synthesis will recover. So we have a reversible system of control of protein synthesis. And it turned out that there were no cues to be formed, although we also did control experiments to show that if we artificially created a cue, we could detect those cues. So the ribosomes, in fact, didn't form a cue under any circumstances, and certainly not when you left the heme out. It also turned out from some other experiments we did that the heme didn't just control globin synthesis. There are a few other proteins being made in these cells, and they were also controlled in the same way by the lack of heme. So it wasn't specific to the hemoglobin. It was a kind of global control mechanism. So I went to New York to study this, and it was a terrific thing to do, to go to find a lab where A, I liked the boss, and B, he was interested in exactly the same things as me. He also had lots of money. Those were the days when the NIH grants were generous, and it was terrific, and I had a very nice fat salary. I was paid as a sort of medical student rather than as a mere scientist, and that was good. And I liked living in New York. But the work didn't really go very well, and there was only me in the lab. And the boss, it turned out, was about to leave and go and set up a new medical school at MIT. So I had to sort of fend for myself. There was just me and a technician who was about twice my age. She was really nice. She was called Grace Vanderhoff, and she was great. But I found myself doing most of the work, the interesting work, at night when Grace had gone home. So I had sort of two projects, one by day and one by night. And at some point, I got so frustrated with this heme project that I decided it would be fun to have something on the side as well. And I thought it would be interesting, and I realized that I could map the genes in poliovirus, and by using the same techniques as Tony and I had developed earlier in Cambridge. And so I went to find somebody, and it turned out to be somebody called Ellie Ehrenfeld, who had poliovirus. And I got some poliovirus RNA from her and tried to translate it, and it didn't translate. Turns out this is exactly the right result. Of course, red cells don't get infected with poliovirus, so they're missing factors that cells that do get infected with poliovirus, and later on colleagues exploited that. But in the process, I sort of thought, well, that must be the case. So what I would need to do is to take, if we know that cells infected with poliovirus are translating poliovirus RNA, so all I have to do is get some poliovirus-infected cytoplasm, add it into the reticulocyte lysate, and it will then translate the poliovirus, and all will be well. But what actually happened when I did that experiment was that protein synthesis stopped. And that didn't happen if the cells weren't infected with poliovirus. Normal HeLa cell lysates didn't have any effect at all. So there was something in the poliovirus-infected cells that inhibited protein synthesis and inhibited globin synthesis. So what was it? Well, this was a very funny time of life, because at that point, Nixon was secretly bombing Cambodia, and a lot of my friends were in danger of being drafted to Vietnam. And it was a very weird sort of political time. We used to have tremendous arguments in the lunchroom. And it almost felt immoral to be doing scientific research when such dreadful things were happening in the big, wide world. But eventually, we solved the problem and discovered that tiny amounts of double-stranded RNA were responsible for this inhibition. When I say tiny amounts, a HeLa cell has about 10 million ribosomes in it. And it turned out that just one molecule of a replicating poliovirus, the double-stranded RNA in it, was capable of inhibiting synthesis by all 10 million of those ribosomes. So it had to be catalytic. There wasn't room on the strand of RNA for all the ribosomes to bind. I mean, that could have been one of the theories. So it must be sort of, that was weird. How could double-stranded RNA make a catalytic inhibitor? And I discovered another thing, too, which had a similar effect. And it wasn't really a very successful postdoc. I mean, the discovery of double-stranded RNA actually that was a Nature paper. In those days, you could publish quite short Nature papers, and you didn't have to have 58 supplementary figures and all that jazz. I think it's a retrograde step now. I think, anyway, let's not go there. But Jim Watson had a very good piece of advice in one of his books. He says, always try to work with people cleverer than yourself. And that, I think, is excellent. I always try to do that. So I went back to Cambridge rather with, you know, I didn't know what to do with myself. And I took a 5-fold cut in salary. That was something quite extraordinary. I'd been on $11,500 a year in 1966 dollars. And I went back to a salary of £800. And I was supposed to pay my national insurance fees, and I didn't realize that. I was self-employed, although I had a college fellowship. And eventually, the Department of Stealth and Total Obscurity caught up with me, and this guy in a raincoat came and said, why aren't you paying your national insurance? And when I told him how much I was earning, he said I was quite right. With the result that my old age pension now is only 2/3 of what it would have been had I been contributing to the pension scheme back then. But anyway. So Richard and I joined forces, and Richard fortunately had a junior appointment. They're called demonstrators in Cambridge, and had a lab. And so and Tony Hunter was still around. And we discovered that it was the same with the heme lac, that an inhibitor of protein synthesis, the formation of an inhibitor was what was responsible for the turning off of protein synthesis, and that inhibitor blocked the initiation of protein synthesis. And Richard and Tony had recently discovered that protein synthesis was initiated with a special methionine-carrying tRNA, and it was the binding of that tRNA which was a step that was inhibited. So that suddenly refocused our thinking. In other words, before then, we'd been studying things in quite the wrong way. We didn't really focus on the inhibitor, and we didn't really know what step in protein synthesis was inhibited, and we knew nothing about the process of initiation of protein synthesis. And I won't go into describing it because at this stage the lab burned down. which you might think was a setback, was actually the best thing that could possibly ever have happened to us, because there was a wonderful insurance policy which said that we could have everything new that had previously been old. And not only that, but we moved out of the old biochemistry department up the road to opposite the laboratory of molecular biology. And the result was that at that point we were actually terribly confused about what was going on. We had a lot of data. Luckily, the 2 graduate students had just graduated and written their thesis, so they weren't affected in the least. We lost 1 paper's worth of data. Everything was burned up. I mean, absolutely everything. The fire was so hot that it even melted Pyrex. And they were these flasks sort of looking like that. Amazing. So, as I say, we went up the road, and it was a teaching lab in a hematology department opposite the famous laboratory of molecular biology. And Max Perutz, who was then the director of the lab, was very kind to us and said that we could use his stores and, perhaps even more importantly, have lunch in his lunchroom, which was run by his wife. And that was fantastic, because the lunchroom had all these fabulous scientists, and you'd sit down at lunch and Francis Crick would come down and explain what he was thinking about. It was nucleosomes at the time, and I got on very well with the people in John Gurdon's lab and had some collaborations. Sidney never went to lunch because he and Mrs. Perutz didn't get along. And so I knew all these people, you know, and not only that, but they as undergraduates, they'd given us lectures in Cambridge. I mean, this was a fantastic privilege, actually. And you realize, as a result, I mean, you know, Fred Sanger, for example, absolutely fantastic scientist, but not a very good lecturer. Rather boring. Even Max was not a very inspirational lecturer. He would come in, and his assistants would bring the then model of hemoglobin, which was a great contraption with wires and things all stuck together with sticking tape and solders and stuff. And he would stick it on the bench, and it was 5 o'clock in the afternoon. He would then turn out the lights in the lecture room, and with a torch, focus on particular, look at this leucine residue, he would say. You know, it's fantastic. It's only 2 angstroms away from... And I just fell asleep. So I didn't really care about that leucine residue and how far it was away from them. Yes, but anyway, but I mean, and so what one sort of got to know was that there are an awful lot of different ways of succeeding in science. And it would be hard indeed to say what these people learn. And not only these people, but look at the list of people down at the bottom who followed on, who were not Nobel laureates at the time. As I say, I got on very well with people, not John, well, I do get on pretty well with John Gurdon, but I was mostly, those days, talking to his students and postdocs. And, you know, they were a quality lot, but we didn't consider ourselves in the same league as the Sangers and Perutzes and the Brenners and the Cricks and so forth. We were just young scientists trying to figure out our particular problems, and we would discuss them over lunch and over dinner and try to help each other and make sensible, intelligent suggestions. And so the previous speaker's point about cross-cultural interchange, I think very, very valid. You know, people would just make random suggestions that were extremely helpful. And you just sort of keep trying and putting one foot in front of the other. And if I had to say what was sort of these people had in common, was that they really liked to understand things properly, and if possible, simply. You don't try to be too clever. When you really understand things, they're usually very, very simple at heart. What looks very complicated at first is actually very, very simple. Anyway, maybe as a result of all this, and maybe also as a result of sort of burning all the previous data, so we had to sort of had a fresh start with fresh equipment, we quickly solved the problems and found this inhibitor was a protein kinase that phosphorylated the initiation factor that bound the initiator tRNA to the ribosomes. And that was a sort of great resolution. And so a problem, I guess this would have been about 1975, so roughly a decade from having first identified the problem, we solved the problem. It was a very good example of a very good problem because it's a clear question, you know, how does heme regulate protein synthesis? The answer is it somehow controls the formation of inhibitor, which is a protein kinase which phosphorylates something, and that inhibits the action of a thing. So we now know that actually there are 4 of these enzymes, and one of the other ones, we actually, I think, really solved it by studying double-stranded RNA and how that worked, but it turned out all the others are the same. And the one we missed was the unfolded protein response. So it was complicated, and the reason why heme and double-stranded RNA, which are 2 incredibly dissimilar things, inhibit protein synthesis the same way is because there are, if you like, receptors for those things which control protein synthesis. And nowadays, this is actually quite a growth industry. I mean, I stopped working on this way back then, and it turns out that the global control of protein synthesis is through this mechanism. And but it presents a bit of a problem. If you're working on a problem and you've solved it, what do? Because it's silly to keep on working on a problem that you've solved, right? Now, of course, there were lots of things you could have done. I mean, we should probably have purified these enzymes and studied them in minute detail and got their 3D structure and stuff like that. But that's really, it wasn't very interesting compared to the main point. So we organized an EMBO workshop to discuss the control of protein synthesis. And I remembered that lecture I'd heard from Borsuk about protein synthesis in sea urchin eggs, and I wondered what was known. I mean, it would be interesting to study. In the case of the heme control, it's really when the heme stops being made, the protein stops being made. It's quite important, that, actually, because otherwise you make a lot of protein that precipitates and you get anemia and stuff. But what was known about the turning on of protein synthesis in sea urchin eggs? And I knew from the literature there was this guy called Tom Humphreys who worked on protein synthesis in sea urchin eggs, and he worked in Hawaii, so we invited him to our meeting. What I didn't know was that he was a very keen cyclist, which I was at the time. And Tom said, "Where can I rent a bicycle?" And I said, "I'm afraid there wasn't at that time anywhere you could rent a bike in Cambridge." So I lent him my bicycle, and as a result of that, we became friends. And what I also didn't know was that he directed the embryology course in Woods Hole, Massachusetts, the Marine Biological Laboratory. And he said to me, "Why don't you come next year and help teach the embryology course," even though I knew no embryology. "And we can do some experiments together on sea urchin egg protein synthesis." So I leapt at the chance and went there. And actually that first summer I learned quite a lot about sea urchin eggs. I also learned a lot about what they didn't have at this lab, which was anything you needed to study protein synthesis. So there were no liquid nitrogen, there was no Revcos, no -80. In fact, all they did was to fertilize eggs of various different phyla and watch what happened for the following week or something like that until you got little swimming embryos. That was nice. Also, there was an Elvis impersonator on the Cape at that time, and we all became groupies and went and listened and danced a lot. And there was also a pub which stayed open and served pizza till midnight, which was very handy because you could sort of just end the lab at about 11:30 at night and then go and get pizza where you're quite hungry by then. So we worked pretty long hours. And then there were lectures starting at 9 o'clock the following morning. And there was a guy called Stan Cohen who worked on the EGF receptor. And he taught me a very important lesson too, which was that it doesn't matter what you're doing in biology as long as you have an assay. And Stan's assay was the premature opening of baby mice's eyes. So he would take his column fractions and inject them into baby mice. And the active fractions were the ones where the mice's eyes opened a day earlier. You know, that blew me away because biochemical assays to me were just things like solutions turning yellow or blue or something like that. And the idea of the baby mice is eye-opening, so it was very useful. Anyway, it turned out to be easy to get sea urchin eggs, and it was also true that when you fertilized them, the rate of protein synthesis went up tremendously by a factor of 10 to 20. This is an actual experiment that I did. And, but the more important thing that these eggs do is divide. And if I fertilize this egg, you'll see what I mean. This is tremendously speeded up, of course. So, but not, I mean, it's tremendously speeded up, but these divisions actually take place at about half-hourly intervals, even though the temperature of seawater is maybe 18 degrees. I mean, this is spectacular. In other words, these eggs are super specialized for cell division. And I went back year after year trying to understand how protein synthesis was turned on and began to wonder about this cell division business. And in 1979, collaborated with Joan Ruderman and her graduate student Eric Rosenthal, studying the control of protein synthesis in clam eggs. Now, clam eggs are a little bit different from sea urchin eggs. You don't see a huge increase in the rate of protein synthesis, but what you do see is the gels on the middle thing, is that before and after fertilization, there's a really significant change in the patterns of protein synthesis. They start making proteins in which we were able to show the messenger RNA was already in the unfertilized egg, but it wasn't translated until after fertilization. And we call these proteins, imaginatively enough, A, B, and C, and wondered what they were there for. And this was Eric's PhD problem, and I thought that was a great problem to figure out how the translation of these messages was controlled. And that summer, oh, sorry, I'm getting out of thing. And this is what a clam egg looks like before and after fertilization, taken by the same kind of microscopy on the left in black and white, on the right in color. And I think you can see, let me see if my pointer can point. Yeah, there's the pointer. Those are the chromosomes. This is the first meiotic spindle. There's one spindle pole, there's the other spindle pole. And this is before fertilization. You can see it has amazing structure, and that structure totally breaks down. I mean, there is a total transformation of the cell by the act of fertilization. And what is the biochemical basis of that? And this was absolutely unknown at the time. And then that same summer, there was a lecture in the one afternoon by John Gerhardt, who was a very distinguished enzymologist and who had become very interested in frog oocyte maturation. I've just shown you clam oocyte maturation. Frog oocyte maturation is very similar in what's going on. It's meiotic divisions. And it had been discovered that progesterone was the hormone that causes maturation. So an oocyte, by the way, this is something I didn't really know. The oocyte is something, the females in the audience will understand this, but I didn't really. The oocyte is the thing in your body which is going to become an egg. And it has to undergo some meiotic divisions and various other processing on the surface before it can be fertilized. So in the case of the frog, I will now show you what it looks like when you add progesterone to the thing there. Progesterone's gone in. This is, again, very speeded up. A white spot forms, and that convulsion at the end is actually the first meiotic division. And what John described was that something was known about this because Yoshio Masui, who was at the time he did these experiments, a postdoc in Yale, had discovered that what progesterone did was to cause the formation of something that he called maturation promoting factor. And the way that he discovered that was that if you take a mature egg, that's the first thing on the right up there, and you suck out a little bit of its cytoplasm, 50 nanoliters or something like that, and inject it into a second oocyte. Now the second oocyte responds as though it had seen progesterone, but much more quickly. Instead of taking several hours, it takes sort of 1 hour or something like that to undergo the same transformation. And so the question is, what was that substance? And what was MPF? Well, it was difficult to say because it was terribly, terribly unstable. And if you tried to purify it, it just went away. And it turned out that, for example, if you added a bit of ATP to the buffers, that helped stabilize it a little bit, but not really. It was destroyed by heating, and it was destroyed by digestion with proteases, so it looked like a protein, so therefore probably an enzyme. And again, I was amazed by this concept that there could be an enzyme that catalyzed cell division. I mean, that sounds crazy, right? I mean, you know, cell division is not a biochemical reaction. It's a much more complicated thing. And not only that, but it turned out that all cells in mitosis have this, and you could assay that in the frog oocytes. It was a difficult assay, but it could be done. And it goes up and down. It appears, and it only appears when cells are dividing. It does not appear in between times. So these eggs are dividing. I wondered, do sea urchin eggs have MPF? And we thought about trying to assay it. And I began to think about, you know, the control of cell division. And I also thought, what a delicious problem finding out what MPF is. And then I did an experiment which was designed to test something completely different, namely, are the patterns of protein synthesis in fertilized eggs the same as the patterns of protein synthesis in parthenogenetically activated eggs? And this was, in some ways, an experiment predicated on my religious upbringing. The idea that you could do a biochemical experiment on virgin birth was quite extraordinary to me. And I liked the idea that you could make sea urchin eggs develop parthenogenetically. I mean, you just had to add a calcium ionophore was the most reliable way of doing it. So I did the experiment, and this actually is not the first experiment. This is a rather clearer second experiment that I did. But I think you can see that the protein labels cyclins. So you label the eggs and you analyze the proteins on a one-dimensional jar. And you can see that cyclin is one of the most abundant proteins synthesized early on, and then it went away. And then it came back again, and then it went away. And we could show that actually for the entire, during these rapid cell divisions, it was coming and going on a regular basis. It's a little bit clearer here. We then went and looked at the clams. Now, Eric and Joan had never done this experiment, so they didn't know that proteins A and B went away. But they do, very clearly. And so from studying the control of protein synthesis, I switched to studying what the hell this protein was, how it went away, and why. And that occupied, I mean, we were very slow and stupid, actually. So, but then you're in a terrible position because you've been an expert in one thing and now you're a complete ignoramus in another very complicated field. And luckily for me, very little was known in those days about the control of the cell cycle. So I read about it, I chatted to friends, I said, is there anything you can see wrong with the idea that this protein which is going away might be part or something to do with the enzyme that catalyzes cell division. And you know, when it goes away, it's because you don't want to be dividing anymore. And nobody could pull any holes in that idea, but they all thought I was crazy. So the point of this really all is DNA and the moment of absolute in life, the reduction of biology to one dimension, Sidney Brenner said. It's a matter of replicating the tape, the program that controls life, and then putting a new copy of the tape into the two daughter cells. And it's worth going back and reading von Neumann's papers on making automata, cellular automata. So that's the cell division cycle. Replication of DNA, segregation of the chromosomes at mitosis, and gaps in between the two processes. So what can one say about that? Well, very little at the time. Lee Hartwell studied the genetics and focused on a gene called CDC28, which controls start, that is to say, the initiation of replication. And Paul Nurse studied CDC2, which apparently controlled the entry into mitosis. And so it looked like they were two different things, and it came very surprising when they were cloned to find that they were interchangeable. But in this, the genetic experiments doesn't tell you anything about the physiology, and there was no hint that proteolysis might be involved. And I should say at this point that nobody had ever suggested that proteolysis was important for the progression in the cell cycle, because it would have been regarded as impossible. Proteolysis is something that occurs in your stomach. If you degrade one protein, you can degrade them all. The idea that you could degrade just one protein specifically at a particular time in the cell cycle, and moreover, we'd established that this stuff, this cyclin, was only unstable at the end of mitosis. It was perfectly stable protein at the other time. So it had extraordinary properties, and we couldn't conceive of it. But that's what happened. So we had to study it. And the answer came from biochemical experiments done by Jim Mahler, who sort of changed the assay and used an unhydrolyzable form of ATP and succeeded in purifying MPF. And it turned out that MPF was a complex between cyclin and CDC2. CDC2 was a protein kinase that without the cyclin had absolutely no activity. And so Paul and I, who discovered that, Paul, I remember wandering around a field in a meeting, and he said, no, it's not a protein kinase. We've tested it, and it has absolutely no activity. And it just didn't cross our minds that our protein could have been an activating subunit, because at that point, only inhibitory subunits of protein kinases were known. I mean, I cannot tell you how bound by prejudice and preconception one is. And, you know, but it's hard to shake yourself out of that until you actually do the experiment and you're forced to change it. It's awfully easy to be theoretical in biology, and you have to be practical. So that was the answer. And, you know, once you understood it, it was really simple. The cell makes cyclin, that turns on CDC2, that catalyzes entry into mitosis. At the end of mitosis, you degrade the cyclin, the CDC2 turns on, and you get on with the business of replication. What was your problem before? But at the time, nobody understood it at all. And so I was incredibly lucky just to stumble on this disappearing protein and to enter a field right at the beginning when nothing whatsoever was known. And I think I should stop there. Thank you very much.
Thank you, Sir Tim. Now we will have a Q&A session moderated by Prof. Thank you.
Please take a seat here. Thank you, Sir Tim, for sharing your life lessons in science with us. It was very inspiring. And to the audience here, please do not burn down the labs to try to get new equipment. Doesn't work that way. So in the interest of time, maybe we can take about 3 questions from the floor. Anybody would like to go ahead? I see a young gentleman here.
Thank you for the wonderful talk. I'm Patrick. I was just wondering, with the burn down lab, would you say that's a stroke of luck, or it's also related to a mindset of turning adversity into opportunity?
I think it was yeah. I mean, when it happened, it was a stunning blow, right? I mean, you know, it was the funny story is that Tony Hunter kept these wonderful lecture notes, and he had filing cabinets full of lecture notes. And we went into the lab the following morning, and he opened the filing cabinet, and supposedly the lecture notes burst into flames, so he shut them again. You know, you just have to deal with it. I mean, it didn't occur to me what a benefit it was at the time, because we just had to focus. I mean, things would happen like, oh gosh, where are we going to get an ultracentrifuge from? So I called up Beckman, and they said, oh, we're going to send 3 of these things to ICI or somewhere. You know, well, they wouldn't mind if you had 1 of them. And so people were incredibly, we had such a wonderful sob story to tell that people were incredibly kind and generous and helpful to us. But it was really that move, I think, which was the even more important thing. But we did have a wonderful new lab and a small lab. And it sort of brought us together too, I suppose.
All right, thank you.
Thanks for the question. I think there is a second question coming up.
Hi, thank you so much for this wonderful talk. You mentioned that you were sort of inspired by a lot of people in Cambridge when you stayed there. And it's hard to not notice that they were all men, for example, and white men also. And I was, just wondering if you could comment on that and maybe give some inspiration to people that do not look like the people you've met and you got inspired from.
I did emphasize that 2 of my collaborators were women along the way. And I think things are changing now, actually. Is Alison here? That they've just appointed the first woman professor in Oxford. Because when I was growing up, there were increasing numbers of young women coming through. It just takes time. I mean, to go through the pipeline before you come out the other end. And it had not been. I mean, I think there was 1 woman lecturer in our department. And there were some famous people. The crystallographer in Oxford, who was the woman who did vitamin B12, Dorothy Hodgkin. So they weren't unknown. I don't think there were any sort of I would have said there was no bias against them, but for reasons, presumably societal reasons, they didn't do it. But this is a very touchy subject, and
Yeah. I'm sorry if the question is a bit.
No. You're absolutely right.
I just wondered.
All those people were men.
Thank you.
And one should ask why. And is it good or bad or?
Yeah, it's open question, I guess.
Yeah. Good question.
I have to agree, this is also a work in progress, and I'm very glad to see that there are equal numbers of men and women, females versus males in the crowd here. Moving on to our third question, and probably your final question.
Thank you very much for your talk and your story. There's something that you've mentioned at the end of your talk, and it's that you felt that you were bound by prejudices, and that kind of you find it hard to break free from what's kind of the standard or the status quo on the field. And I think that's something that comes a lot in a lot of breakthroughs and a lot of things that are holding up great scientific discoveries is these biases. So what's your best advice from your experience to young scientists to kind of break free from?
Well, I guess don't be afraid to think differently. I mean, and but you need the clues, right? I mean, you need to be drawn in the right direction. And it's so important to keep your feet on the ground and not go further than the actual evidence. But it does help to dream. I mean, one of the things that in retrospect, I find very hard, a lot of the pioneers in this cell cycle field were really, really fine scientists. I mean, really clever people. And nobody ever thought about this. So it wasn't just sort of, it was sort of collective blindness, collective biochemical blindness, just from looking at an example, a lone example. I mean, we should have known better.
Thank you.
Okay, the last question over there.
Hello, I'm Ram. You mentioned also about the prejudice, but I'm interested in validating the results, because you mentioned that science are putting you in that direction. Yeah, that breaks the prejudice that you have. Were the results that you have before did you try to ask someone to try the same experiment and led to the same result? Because sometimes we get a failure, and just sometimes we just doubt ourselves if we're doing the experiments right. Right?
Well, let me tell you, a point I didn't make was that this was a really bad research project in one respect, which is that the material, sea urchin eggs, are only available for 1 month of the year. And so you could only do a month's worth of experiments, and then you come back. And I was terrified. I'd come back the following year. Would it still be true? So the very first thing we did was just to repeat the experiment. And I was so relieved when the protein still went away a year later. And of course, we tried. I mean, one of the things I didn't mention was that, first we did this with sea urchin eggs, and then we wondered whether it happened anywhere else. And I persuaded a student in the lab, this was a summer course, whose project wasn't going very well, that he ought to look in clams to see whether anything went away in clams. And we were delighted when we found that these 2 proteins went away in clams. So we were pretty confident that, it wasn't an artifact. And in any way, the way the experiment was done, it was hard for it to be an artifact. You couldn't really be fooled. It was a very robust experiment, and I was very used to doing that kind of experiment. So I sort of, those things that I showed you, I was very good at reading them by then. If you're not, they wouldn't mean so much. And indeed, I slightly congratulate myself. I think, I do regard myself as a bit of a fraud as a Nobel laureate, because all I really did was to see this protein go away. And the first time the protein went away, it didn't really go away terribly convincingly. And nevertheless, I don't think anybody else would have noticed except for me. Put it that way.
Thank you very much.
Thank you very much, Prof. Tim.
Thank you.
Thank you.
Thanks for listening.
Thank you, Sir Tim and Prof. Our next speaker is Sir Andre Geim, who will share about the random walk to graphene. Sir Andre, please.
I have to zoom off. It's a shame, but I would love to have your Okay, so right, switch of subjects from life scientists to material scientists, and during the next 30-35 minutes, I'll try to tell my story, why I'm here and how did I get it. So when someone gets a prestigious prize, especially something like Nobel Prize, there are usually 2 questions asked. How and why? How, I'll tell you story behind, and why it usually sounds, why this guy, why not myself? So I'll try to explain why not me, but this material, which is called graphene, deserves the Nobel Prize. Hope to persuade you by the end of the talk. My story probably will start in somewhere middle 90s, when I was a young tenured professor in the Netherlands at that time, and getting tenure is kind of relaxing. You just can forget about your rush for publications. You have secured your position in the world, so you can think about something for your soul. And I was thinking about what else can I do. I'm so-called quantum condensed matter physicist. I was doing some very obscure experiments, which I got grants, I got PhD students, projects, but at the same time I start doing something else, like a hobby. And later, those kind of experiments were called Friday night experiments, which comes probably from the fact that on Friday night we had a lot of beers and decided, okay, what else can we do? Something interesting, something outside the well-trodden path. One of the first experiments I have done was using the magnets which we had at that time in the lab. Nothing spectacular with those magnets. They were pretty high, up to 20 Tesla, but there were many labs in the world who have those magnets, and they consume a lot of energy, cool down with the hot water because its current goes through a copper coil, essentially. People, including myself, usually put some solid-state little things, semiconductors, and studied what happens in high magnetic field and at low temperatures. Rather obscure experiments. I was doing mostly mesoscopic superconductivity at that time, if this tells you something. And I wanted to something easy, something out of normal way of doing things. And the question I asked, what happens with water if we put water in high magnetic fields? It was a random question, but not so random at that time, because I also had some idea what's happening, some background knowledge what's happening with water in small magnetic field. No time to talk about this one. So I thought, okay, we have those facilities here. Why wouldn't we try to study water in those highest magnetic field, essentially static, available on this planet? So once upon a time, I took literally a bottle of water, pour it inside the magnet, and this is, you know, not the way usually people do with their relatively, or I would say very expensive equipment, pour water on top. And I'm not sure what I expected out of this experiment, and actually why I have done this way. Probably beer were partly to blame. But the result was spectacular. So what you see here is a water ball of the size of a tennis ball, which freely floats in the air, and green, it's a laser light which shines it to be better visible, and those droplets on the wall kissing this ball, and this continues until you switch off the magnetic field. Well, I certainly was rather well educated already at that time, so I certainly didn't believe that I invented an anti-gravity machine. So immediate culprit for this levitation came as diamagnetism, and after half an hour of calculations, I did find out that diamagnetism is enough to explain the phenomena. Diamagnetism is a quite well-known phenomena, 100 years before, discovered, I think, by Kelvin, who noticed that if you have a strong magnet, even permanent magnet, it produces some repulsing force on seemingly non-magnetic substances. We usually attribute magnetism to something like iron, nickel, and those magnets, but you, paper, everything around us, tables, those things which you consider non-magnetic, they're in fact magnetic. So scientifically, it was not surprise. But what was surprise, really, for me, was that when next morning I came into the audience, I found out that, not the audience, to the lab, and tell my colleagues who professionally was dealing with high magnetic field for since their essentially PhD studies and about to retire, they didn't believe this. And I have to go back to the lab and show this phenomena to them, and then show other colleagues, and then other colleagues came back to the lab from overseas want to see this. It was a lot of hype around, and all colleagues came with beautiful ideas what else could be put in those high magnetic fields. So about a couple of weeks later, I was so fed up with those experiments, so I decided that I need to do something and finish with this circus which was going around essentially all the time. And the idea was to put a frog in magnetic field. If everything magnetic, you need to show we are magnetic. My face didn't fit inside the magnet, but the frog did, and I managed to make a short movie of the frog. Probably the hardest experiment I have ever done, because condensed matter physicists I do with things which do not jump and do not stick to my fingers, and frogs are not. So these days, whether it was worthwhile, I got Ig Nobel Prize for this work, and I agreed to accept this Ig Nobel Prize. Could reject, and no one would know, but this. Sometimes I go to a conference, say, American Physical Society meeting or something like that, and people come to me, oh, I know you, and I roll my eyes. I have no idea what graphene is, but your frog. That's kind of happenings, and I particularly cherish the letter I got about 25 years ago. The letter reads, as you see, I'm 9 years old and I want to become scientist, want to learn how you float the frog. So sweet from the girl from Alaska. Sometimes random thoughts and random experiments, as I try to persuade you, they do work. So the next random experiment has nothing to do with my condensed matter experience, was Gecko. And I learned from the background how geckos stick to the walls. They don't use glue, they don't use anything like that. So so their toes covered with tiny, tiny hairs. And if you pull out one of your hair and you touch any surface, there would be a small attraction force, which is called van der Waals force. Sometimes it's mediated by condensation of water, but doesn't matter. It's van der Waals interaction. The force is minute, nanonewton, but 10 nanonewtons, something like that. But geckos learn how to use all their hairs, and there are billions of them on those toes. Each one produces 10 nanonewtons, billions produces 10 newtons, and it's quite enough to keep geckos on those. So the idea was, all right, I'm condensed matter physicist, but this is standard size, what I'm usually doing in my lab, dealing with that. And using the facilities which we had, we created this forest of hairs, which you see they can stick to each other, they stick to other surfaces. The original idea was to make a gecko glove and put one of the students out of the window with this gecko glove. Not for the lack of students, but for the lack of the material. It has never happened, but eventually I have to limit myself with this toy model to demonstrate the works. It turned out to be quite prolific paper, a couple of thousand citations, people still trying to do this. I have seen a real person hanging from the ceiling using the same kind as this gecko tape principle, but unfortunately they who are dealing with this still didn't learn how detach and attach again there is climbing going around like on this picture, and after a couple of attachments it no longer works. So I gave you an idea what kind of mentality it is doing random experiments. So now coming to the subject of the Nobel Award, it was also originated with kind of an idea how thin graphite can be made. Why graphite? Because this material is relatively easy to split. I also know at the moment that people are interested in carbon materials like carbon nanotubes and so on, and graphite itself interested, but no one managed to make thin films of graphite at that moment. And I thought it would be interesting to see how thin you can make those films. So the idea was essentially the same as ancient Greeks had. You know, they invented the concept of atoms by just saying you divide a stone into two, then in two, then in two, and so on, until you can't divide it into anything further. That's an atom, the smallest particle it is. So we start dividing graphite into smaller pieces using different technique. It didn't work for a few months. My PhD student, whom I gave it idea, did manage to succeed in anything new until we tried a sellotape. So using sellotape, you stick it to the surface of graphite, you peel it off, you have a fresh surface of graphite. Thousands of researchers around the world did this kind of approach, method of cleaning graphite, putting tape on top, peeling it off, throwing the tape away, studying graphite. What they didn't realize, that throwing away the tape, they possibly had thrown their Nobel Prizes as well. So what we did, we look at this tape in a microscope, what I did at some moment, and putting, this is a typical image what you see. If you shine lines through, the light goes from behind the screen, and you see on the tape under some magnification there are thick pieces. What thick non-transparent pieces tell you? That material is a metal, it's not transparent. But when you see a metal which is transparent, you immediately get an idea that it's a very, very thin metal. Looking at this metal is here. This piece of graphite is only a couple of nanometers thick. And this was kind of eureka moment for me. I can make graphite really, really thin. So why it was a eureka moment for me? Because, yeah, education helps. Sometimes you just see something and throw it away without noticing. What I knew, and you know this, and let's learn it together, that things around us in the world where we live are all three-dimensional, have height, width, length, and so on. And why it is so? You wouldn't find anything which is a wire which is one atom thick, or a single atom thick sheet. Why? Not because you wouldn't be able to see it, but because things do not go like this in our universe. Because whenever you make a material, to grow this material, one way or another, you have to have temperature. And temperature causes atoms move, vibrate, and so on. And if initially you get something which looks a bit flattish, like you will see somewhere now here, then eventually it's all merges into three-dimensional architecture. Or you can say a different way, the nature try to utilize all three dimensions, and this quenches those violent vibrations which destroy the order. So whatever you try, you can't grow two-dimensional materials, which are one atom or one molecule thick. So and this was surprise that we could make those materials which assumed by all those many theorems impossible to produce in our three-dimensional world. But we played a trick with this Scotch tape. Essentially, we tried a trick, which is taking three-dimensional material, it's graphite, or you can take any other materials, and imagine that you have very tiny tweezers and you pull an individual atomic plane out of this three-dimensional material. Those theorems, physics theorems, which you can't break during growth, you can still do in this experiment, because what you do, you start with three-dimensional material, which was grown at very high temperature, and then at low temperature, which is relatively low, it's our room temperature, you pull out an individual plane, and there is no theorem that forbids it. But the mentality was not there to do this kind of experiment, and even thick for two-dimensional material. So this is an example of a photograph at that time, about 15 years ago, 1 millimeter monolayer carbon, one monolayer, one atom thick carbon crystal, which is seen here. You can see it by naked eye. Also quite a surprise, because one atom you just think it would be the thinnest possible film you can imagine. You expect it to be completely transparent. Still, you can visualize. Of course, you can use all these powerful modern techniques, including transmission electron microscopy, and make sure that it's indeed crystal of carbon atoms. And this is real photograph there. Okay, so what? We make those monolayer carbon sheets, extremely thin structure, and one say, so what's so special? It's good, it's nice that we can extract atomic planes, but so what? And if it would be just this trick of extracting atomic planes, it would be probably, I wouldn't be here. It would be just a scientific curiosity. But then, over, say, 3-5 years, we start looking at the properties of this material more carefully and found out that it's extremely unusual. It's turned out to be not only the material with the largest surface area, 1 gram covers a few football pitches as we measure surface in Manchester, but this kind of expected from the thickness of the material. What we didn't expect, that it turned to be the strongest material, at least in the lab, ever measured. It's not only strong, it's stiffer than diamond, but try to pull diamond or compress diamond, you wouldn't do anything. You would be not able, but this material is pliable, is stretchable, this graphene, very unusual properties. And this shows record thermal conductivity, it's outperforming diamond, and it also carries current 1,000 times per thickness higher than copper or gold or silver can do. And there are plenty of other properties. But what we mostly learn from this example of this material, that if you take one atomic plane out of three-dimensional material, you find out that properties are completely different and unexpectedly different from its parent. Then from those properties, new science did came along, and I have no time to describe this science. I'll give you a few examples. One probably most important example is what the Nobel Prize was given mostly for electronic properties of graphene. Bear with me for 2 minutes. It's a rather complicated transparency, but I'll try to give you an idea. When we describe materials which are around us, metals and semiconductors, we use so-called Schrödinger equation. It's it's written here. It's just like bullets move with certain mass and so on. But in the world of astrophysicists and particle physicists, when particles move with the speed close to the speed of light, you use another equation, which is called Dirac equation in simplest form. And this is essentially relativistic versus non-relativistic physics. What graphene bring about, it brought mimicry. In our normal world of speeds slower than speed of light, electrons within graphene behave like they behave near neutron stars during the Big Bang, if you wish. So you can now mimic with graphene many experiments which was impossible to mimic before. And this was, for many years, it was the focus of a very large community who were dealing with this. I'll give you one example what kind of phenomena you can address with graphene. One of those is called Klein tunneling, predicted 80 years ago or something like that. And it's about the following. You know, if you drive your car into the wall, what would happen essentially. If you ever heard about quantum mechanics, you know about phenomena of quantum tunneling. And according to the laws of quantum world, if you drive a quantum car into a barrier, some ghost of this car, depending on the height and thickness of the barrier, would go through. That's what usually physicists are dealing with. But if you live in the world governed by the laws, particles, electrons behave within graphene, then if you live in this kind of world according to those laws, then if you make a car and make a barrier, however high or however wide the barrier is, the car can go through without noticing this barrier. This is very important phenomena. It's short of time, so let me skip several other examples of phenomena which we observed over years graphene, and just give you an idea that the search for new phenomena in this material goes on. It's last year example. A rather simple experiment we have done, and it was astonishing. More about chemistry rather than physics. You know what the philosopher's stone is. Philosophers try to find a substance which turns something into gold. So in our case, this something is electronic waste, where your computers, your mobile phones go into this, where that's our rubbish. You put aqua regia, a mixture of two acids, wash realistic this kind of electronic waste, you put it through, you get a liquid, and then you pour this liquid through a collection of graphene flakes. And this is real-time experiment, and you see graphene which was black, plenty of these graphene flakes there, getting yellow and yellow. Then you take this yellow stuff, burn it literally in air, and you get gold, essentially. Yeah, so it's last year experiment. It still remains to be seen whether it would go to industrial production, but it's very efficient way of extracting gold. For me, graphene was dead already 3 years, scientifically speaking, because we knew everything about graphene after 3-5 years. 1,000 people around the world studied this material, so nothing left there. But somehow, as you have seen from the previous example, we're still studying graphene. And the reason for this is this turns out the materials with scientifically incredibly long longevity. Why it is? Because this material reincarnates itself in different kind of subjects. The very first one was quite obvious. A year later, after extracting graphene, we tried the same trick with the Scotch tape or many other materials and showed the world that it's not only graphite can be pulled down to individual atomic planes, but many other materials as well. Now we know hundreds, if not thousands, different materials which can be atomically seen, from three-dimensional counterpart to two-dimensional counterpart. Before graphene, no one interested, no one even tried to look through those materials. And imagine when you get hundreds and thousands new materials, people are trying to find interesting and useful properties of those materials as well. What else you can do with graphene? A next probably 3.0 or 4.0 incarnation of graphene. If you have a library of those materials, you can do the following and make by design layered structures and ask yourself what properties would be. How can you use those properties? It's like Lego game, but on atomic scale. You can make materials which nature can't do at all. So we and other people are intensively doing this today. It's including Singapore. Many people are working on these materials, and this is real. This is not cartoon. This is an example of new material on the right, where you can make material by design stick in those atomic planes. The latest one, another incarnation which we inactively science, and gives you an idea how it's important to change a little bit mentality. That's what we have done previously, 15 years ago, 17 years ago. We extracted graphene and forgot about graphite. Change your mentality. Forget about graphene. Ask yourself the question, what happens behind? And what happens behind is a cavity, one atom or a couple of atoms of thick cavity, essentially two edge dislocations with an empty space. Interesting to study how atoms, how liquids, how gases go through this cavity, what properties would be of confined liquids, gases, and other materials within this cavity. And this is all real. We can do this on the lab using so-called van der Waals technology and putting this kind of spaces, which are usually graphene spaces in between two atomically flat systems. So plenty of stuff still going on. Graphene continues to reincarnate itself in through different sisters and brothers, one atom or one molecule in material. So graphene essentially, during the last 15 days, strongly affected and sometimes reshaped the whole fields in physics, chemistry, and even penetrates these days into life sciences and so on. It's already in many industrial products also. They rarely call graphene, they just improve properties of the material. My last slide is more or less philosophical for the youngest people in this audience, for students, PhD students, and so on, who might sometimes be disappointed how their PhDs and so on work, that everything was studied in physics and in energy. So that's a philosophy goes like this. Take a pencil, draw a line, like here. What this black stuff is flakes of graphene, which comes from the pencil lead, because pencil lead contains a lot of graphite. It's the basis material. You can do this. Take a microscope, look carefully at this pencil trace, and like in Scotch tape technique, if you have enough patience, and I have done once, you can see thinner and thinner pieces in this pencil trace. And the thinnest one, you can find even graphene about this place. Pencils and graphite known for 700 years. So for 700 years, graphene, essentially was under our noses, in front of our eyes, whenever we draw something by the pencil. But it took to 21st century having facilities which we have there to realize that graphene, such a wonder material, is underneath. So that's probably tells us something that we don't know yet everything about our world. Thank you.
Thank you, Sir Andre. We will now have a Q&A session moderated by Prof. Lo Hongyi. Prof. Lo, please.
Thank you, Sir Andre. That was a really inspiring talk. Please, there's water.
Yeah, I wonder what is.
So let's open up the floor for questions. Any question from the audience first? If not, we have a couple of questions from the Slido. There was a questions that actually got missing, but the question was, what is your next plan for your Friday night experiment?
We're essentially doing it, these cavities which are very thin, it came as a Friday night experiment. It's required to change in mentality. It's, you can call this kind of relevant to graphene, but the idea is the same kind of using tricks, trying to find something which is not directly related to your normal line of work, something outside, something which you have never dealt before, like molecular transport and liquids, solids, I have never dealt before. But this kind of experiments are now done because I have facilities. I can do these van der Waals heterostructures, I can do this stacking. So in these Friday night experiments, mentality is the same, but facilities are now based on what I have around, like flying frogs. I wouldn't do this experiment with flying frogs if there would be no magnets around. So now I have this huge range library of two-dimensional materials. I'll try to play with them. The idea is the same, trying to play, doing your work.
Thank you. That's like your playground, isn't it? Question from the floor.
Good afternoon. I'm Chao Yang from the University of Hong Kong, and I'm focused on the environmental engineering using 2D materials. So since the graphene has been discovered for 20 years, and now there are many commercial products in the society or in the industry for battery or some energy storage. So for environmental applications, we also focus on the our lab also focuses on the research study and the commercialization of the graphene for water treatment. So my question is, what's your suggestions for the young researchers for the study of the research and the commercial products respectively for different aspects for study on graphene?
You know, to be honest, I'm not an industrialist or a property developer, so I'm a professor doing mostly physics at the university. So to give you advice how to do commercialization of the products, it's not my area of expertise. So all what I can say, try. We know that two-dimensional materials, especially graphene, nowadays are in many commercial products, hundreds and thousands of them. One has to try kind of adventurous stuff. Don't be a follower, but try, even if you're a follower, try to jump somewhere a little bit away, so from the routine which you suggested to follow. So that's only be more adventurous in your research. So that's all what I can say. And about commercialization, I can't say much.
Yeah, for the research, my question is, we try to discover new 2D materials or some as inspired by you, some the new discovery, new discussion, such as the graphene, such as the biolayer or different 2D materials assembled to the layer for different. So these two different directions. So how about your suggestions or comments on the discover new 2D materials or some new phenomena in the graphene-based materials?
I have a colleague who would answer your question. If I would know what to do, I would do it myself and wouldn't tell you.
Yeah. Okay, thank you.
There are 3 people lining up there for questions. So let's turn over to the other side of the room. Please go ahead.
Thanks a lot for your interesting talk. I'm Vishaka. My question is, will graphene replace the silicon-based electronic industry? How long it will take?
Silicon industry, how long will it take? That's a tough one, huh?
There was a lot of discussion 10, 15 years ago about possibility of what you are saying, replacing silicon. First, it was over-optimistic, always. I never really believed that it's possible because silicon is so wonderful material and it's such a huge trillion-dollar industry. It's very hard to do something. But when other two-dimensional materials came around, they offer a range of properties. Some of those are semiconductors, unlike more or less metallic graphene. So people are still, there are papers in Nature. Recently, Huawei announced that in 5 years they will be doing integrated circuits with graphene, but I don't believe it's a question of 5 years. I think it's a few decades, but many other two-dimensional materials, molybdenum sulfide or other dichalcogenides, they do offer something what silicon can't do. It's much smaller, transparent electronics, which would come eventually, but not in a few years. In a couple of decades. It takes a long time to progress from academia to consumer products. But we're looking at the moment on very simple products and not in semiconductor industry like paints, like tarmac enhanced by graphene or concrete saving enormous amount of carbon emissions, something like that. Very simple solutions are based currently. We have to wait for more complex solutions.
Thank you. What's your opinion about mica, 2D mica?
Mica is one of many, yeah. So we also dealt with 2D mica, yeah. It's an interesting material, good, for example, for proton transport through this one. Very hard to make 2D mica in large quantities. If you can make 2D mica sheets like graphene square meters, it would be really interesting for many applications. But as you know, we can only Scotch tape mica these days, but it's an interesting material, but one of dozens of interesting two-dimensional materials.
Thank you for the question. I'm afraid we should give chance to one more question. We have a minute and a half left. Please go ahead.
Hi, I'm Taya. I was wondering, you told us about your successful Friday night experiments. Could you tell about a failed experiment, or about an experiment where it seems like you're asking kind of very open-ended questions, and where you start experimenting, you're like, okay, well, this isn't really going to go anywhere?
If you explore your Friday night experiment, it leads you to failure. That's pretty much the question. What do you do next?
Yeah, it's a good question, actually. One thing I noticed, and I noticed in my written novel lecture, that you expect, like, you know, in venture capital, people expect now 9 out of 10 projects fail. What turned out with me is that nearly half of these projects, which were starting with simple ideas, brought to something interesting. So the chances of success, which I don't understand, are incredibly high than usual. But of course, it's a random idea, sometimes stupid idea, and stupid ideas must fail. And what you do, that's the hardest question, which actually everyone at a stock market, financial stock markets, knows when to cut losses. And that's really, really hard. And yeah, you tend to carry on, but at a certain moment it doesn't work. It's really tough solution. But to cut losses, I sometimes say my PhD students, we need to cut losses. And say, oh, can we measure another month? Yeah, very tough.
That was a great advice. I'm afraid we are just on time to end today's sessions. Thank you again for the very inspiring sharing.
Okay, thank you. Thank you.
Thank you, Sir Andre and Prof. Loh. If you're keen for more exciting research presentations, do join the online engagement by TUM Create on CellFACE Imaging-based flow cytometry for hidden haematology biomarkers on the Give Me platform. For now, we will adjourn for a quick break, and please be back here by 3:25 p.m. for a panel discussion. Thank you.
Welcome, everybody. My name is Ulf Schlichtmann. I'm a professor at Technical University of Munich in Munich, Germany, and I'm also serving as the CEO of TUM Create. TUM Create is a research platform of TUM in Singapore at the Create campus of National Research Foundation. TUM actually has been active in Singapore for more than 20 years, first with a teaching subsidiary, TUM Asia, which is still going very strong. And then when oops, that's a bit too quick. When Create was started about a dozen years ago, we joined as one of the first 3 university partners of Create. In the first 5 years, 2011 to 2016, we tackled the topic of electromobility in tropical megacities, which at that time was very innovative. So we actually built a working taxi from scratch, which you can see here. Then in our second phase, the next 5 years, we focused on how to optimize transport in megacities from a systemic perspective. And every 10 years, the Create entities reinvent themselves. So now, since last year, while we're still pursuing some smaller projects in energy and mobility simulation, we focus more on life sciences. Our major program right now is called Proteins for Singapore, where we are working on alternative ways to solve the food crisis which the world is facing and the sustainability crisis. And we have another very exciting project which we want to focus on today, which is called CellFACE, which is a novel way of diagnosing blood. And this is going to be the main event. And for that, I would like to hand over to Qianyu and Kerem, 2 research associates with TUM Create, who are working on the CellFACE project and who will now give you an introduction to this project. And then we hope, even though we're a bit late, that we still have time for your questions. So, Kerem and Qianyu, the floor is yours.
Thank you. Okay. Thank you very much, Professor Schlichtmann, for the nice introduction. And also thank you very much for GYSS committee for inviting us. We will today talk about CellFACE project, which is one of the ongoing research project under TUM Create. We are focusing on developing a new next generation imaging flow cytometer for revealing some hidden biomarkers which could be potentially used in clinical diagnosis for various diseases. So basically, first, maybe I can talk about who is involved into CellFACE project. So we are a highly interdisciplinary project from various industrial partners and academic contributors. So core team is coming from Technical University of Munich. We have clinicians on board. We have data analysis and AI experts. We have also contributors from NTU for developing microfluidic solutions. Also, we have contributors from AUS and AUH for clinical parts. And for the TUM Create research team, right now we are 3 people in this image, but we also have a few more team members till this picture was taken. So my name is Kerem, the person on left. We have Qianyu. She will also introduce you data analysis part, and we have Komal in biomarker analysis part. Basically, the vision and motivation of CellFACE project is actually developing a low-cost, label-free, and quick blood analysis. Because actually, if we look at the recent technology, gold standard technology used in clinical setting, so basically, even though this is quite effective, there are still room to expand. First of all, cost is still a problem. Secondly, label-free analysis are still something that we need to explore. Because, for example, in the case of hematology analyzers or flow cytometers, labeling procedure is still a very labor-intensive and costly process. And thirdly, since we are not only analyzing signals, which is the case in flow cytometer, for example, but also we can provide cell morphologies, which is actually a unique way because we can directly have a look at cells and then we can detect some abnormalities, which may indicate various sorts of diseases. So at the end, our aim to actually develop some panel, some biomarker panel that can link various cell types, populations to diseases for quick and low-cost diagnostics in clinics. So what we are focusing on is actually fever. So fever is actually something we all experience in our lifetimes. But the thing about fever is that it's a very broad, very general symptom. So there are various reasons relying underneath. And this could be simply, for example, when you get a COVID booster, most probably you will have fever because your body responds against a kind of foreign agent. So that's why, for example, you will experience fever. On the other hand, for example, if you have some kind of viral or bacterial infection, still you will have fever. This could be the case in cancer, pneumonia, or maybe some kidney diseases. So again, fever is a very actually basic but also important symptom that we need to address. And actually discriminating the resource or the cause actually has a very, very huge impact in clinical setting. So just to give you some idea, some numbers, actually the 5% of adults and 15% of elderly people, when they apply to emergency, has fever. So fever is actually the second most common complaint among these misdiagnosed or somehow diagnosed, but a very in a long term. Because the investigating fever and finding the exact cause is quite time-consuming and costly process. So that's why actually, if we find a way to diagnose the underlying reason in a fast, in a low-cost manner, actually this could potentially have quite a huge impact in patients' welfare and time. So investigating fever, actually, traditionally, we use blood count, whole blood count. So basically, blood provides us quite a nice overview what's going on in the body because we basically have a look at three distinct type of blood cells, but also we can further analyze, for example, subtype of white blood cells. And in some cases, for example, in leukopenia, which can be caused by viral infection, we see some white blood cell numbers actually increases or simply fluctuates. Or in, for example, the case for leukocytosis, neutrophils can actually fight against bacterial infection so that it also gives us some idea what's going on in the body. But still, actually, these are quite general, broad, maybe indications. And we need something more specific to address what exactly going on in the body. We need some more specific biomarkers. So if we have, for example, look at some blood smear, which is the gold standard for, I guess, for last 150 years, actually, we can also see the variation in number and morphology of cells, which is actually quite a good indicator. But problem with blood smear is that, first of all, this is super labor intensive. It takes time. And secondly, we can only analyze a very small amount of blood. So again, microscopic analysis has been done for quite a long time, but we need something more specific. That's why, for example, flow cytometer has developed, and this actually changed the life science and medical sciences quite a lot. But still, the problem we are facing is these technologies cannot really provide us a low cost and label-free fashion analysis. For analyzing cell morphologies can tell us some abnormalities, in morphology and population size. And also we can link these information into different diseases and different conditions. Oops, sorry. So for further investigating fever, we need to actually differentiate the source of the fever, which could be viral, bacterial, or just simply fungal source. So the problem here is that the discrimination of viral and bacterial infection requires quite a intensive work. That's why actually clinical diagnostic takes some time, which may result delayed diagnosis and delayed treatment. So in the other, for example, there could be other cases like non-infection cases, autoimmune diseases, malignancies, some cancer diseases, some allergy or something like this. So that's why we need to actually differentiate the source. And again, as I mentioned, actually right now the gold standard in clinical setting is using hematology analyzers. So basically hematology analyzers are very good at providing an overview about blood cell count. But the problem here is that they cannot provide, for example, cell aggregates, for example, leukocyte aggregates or platelet aggregates, which might be the case in different types of infections. This is something we need to investigate because these machines can only provide us signals, but not morphology. So as a result, actually, there is a unmet clinical needs that we need to deliver label-free, low-cost, and point-of-care solutions, not only for general healthcare, but especially for emergency department and intensive care. So the processing should be minimum, should be label-free manner, and it should be repeatable. So what we also introduce is actually we can analyze using artificial intelligence so that we can be more precise and quick. And from here, I think Chenyu will take over and then give you a little bit more about technical aspects.
Thank you, Karen. So now I'd like to talk more from the data analysis part. From this slide, we can see that once we receive the samples from the hospital, we will inject the sample through the digital holographic microscope and do the imaging. Then we're going to measure the blood samples, record the images, and do the analysis. And then from the right side, we can see some examples of the aggregate, like leukocyte aggregate, platelet leukocyte aggregate, or just platelet aggregate. Then in our next slide, we can see from the left side, in this video clip, we get the idea how the DHM microscope record the high throughput blood sample images. And from the right side, in TUM, we have already successfully differentiated 8 different leukocyte subtypes. In the top row, we can see the reconstructed phase images of leukocyte subtypes. And the bottom row indicates the corresponding heat maps of the top row phase images. And here the scale bars are 5 micrometers. So we can move to the next slide. Here we can see the plot of percent. We can see the by applying the deep learning algorithm, we feed the large scale of cell images to the neural network to train the model, and then to perform the object detection, the instance segmentation, and distinct cell types classification. And our target is to find out rare aggregates and quantify them. So with these analyzed statistics, we are able to provide to clinicians to support them for diagnosing the severity of patient diseases. So nextly, we maybe Karen can take over the slide.
So we would also like to talk about few use cases that we already worked on, which one of which is actually identifying abnormalities in blood, and this might indicate actually leukemia. So for example, for the first figure from A, you see that healthy control samples, this shows quite a 3 different actually population. And when, for example, for acute myeloid leukemia or lymphocytic leukemia, or for example, chronic lymphocytic leukemia, you see actually these populations shift and a little bit more intense. So definitely there is a difference compared to healthy control. So basically just getting white blood cell count from DHM, we can discriminate these diseases. But for example, myeloproliferative neoplasm, the characteristic didn't change much, but still, for example, we can further analyze these data population through our artificial intelligence network to come up with more precise results. So further, maybe Jane, you can continue in this part.
Yeah, thank you. Actually, we can combine this slide together with the next slide. So this is here, this plot, we can see the percentage of platelet aggregate of COVID-19 patients in ICU at the very first measurement day. And it's clearly observed that the mean value of thrombocyte aggregate percentage is lower than the mean value of mid and severe patients aggregate percentage. And actually in the second slide, it also depicts the amount of thrombocyte aggregate percentage at the maximum measurement day in the relationship to total thrombocyte count from the different cohorts. And these 2 plots state that our label-free technology can greatly helpful for clinical diagnosis to identify whether this patient is infected or not. So yeah, that's all for our this technology from the
yeah. And for example, so far we showed you a few applications, which one of which could be whole blood count, for example, for leukemia in particular, and other is, for example, is a very recent study that we did in Munich site actually, the COVID study. So all of which actually showed us that CellFACE can provide us a very unique way to deliver some novel diagnostic information. Because, for example, if you have a look at the charts here, even though fluorescent flow cytometer and hematology analyzers are gold standard in both academia and clinical settings, these technologies actually require some labeling procedures, the training personnel to use it, and these technologies, for example, cannot provide us cell morphologies that we already showed you. These are unique and important information for diagnosis different diseases. On the other hand, for example, we have regular traditional biomarker analysis, but these actually analysis can fluctuate a lot, especially in very severe infections. And for, as I mentioned, blood smear analysis is very actually good for determining, analyzing cell morphologies, but this is a very label intensive and very kind of limited way to analyze blood samples. So in the intersection of all these, actually what we propose is providing a label-free, low-cost, and hopefully a technology that can provide us a unique and new biomarkers.
So thank you everybody for joining. I hope this has given you some overview of TUM Create and CellFACE, and please do feel free to reach out to us for if you have further questions. We'll be very happy to answer them. Thank you everybody.
Yep, thank you very much.
Hope you have settled down and have a short break. This afternoon we have a panel discussion on revolutionary tools for science. The panelist members are Prof. Ciechanover, Prof. Didier and Sir Konstantin Novoselov. Moderating the panel today will be Prof. Gisbert Schneider, Director of the Singapore ETH Center, SEC. Please warmly welcome the panel and our moderator, and I'll hand the session over to Prof. Schneider, please. Thank you.
Thank you very much. I'm happy, it's a pleasure to chair this special panel this afternoon on revolutionary tools for science. And as you will see in the future, I have found one particularly revolutionary tool, that is my glasses, because this way I can see you and that way I can see my notes. Let's kick it off right away with a statement. It's a general perception that new methods and tools are essential drivers of scientific breakthroughs. New methods and tools are essential drivers of scientific breakthroughs. Today, we want to explore this hypothesis. And with me, I have three fantastic panelists, revolutionaries in their own right. Aaron Ciechanover, a professor of biology, medicine. He won the Nobel Prize in 2004 for characterizing the method that cells use to degrade and recycle proteins using ubiquitin. Welcome. Next one is Professor Didier Queloz. He was awarded the Nobel Prize in Physics in 2019 for the discovery of an exoplanet orbiting a solar-type star. And next to me, Sir Konstantin Novoselov. His work on graphene earned him the Nobel Prize in Physics in 2010. So we have a diverse group of panelists here with me, and they all work with various kinds of tools and technologies, be it large-scale facilities or very small-scale tools in their daily work and laboratories. And we will explore to which degree these tools are essential for scientific breakthroughs and how to define and identify new revolutionary tools for science of the future. Now, but first I would like to ask all three of you to briefly introduce your field of research and maybe mention one or two tools that have been essential for your seminal findings in the past. And I'd like to invite you, Aaron, to make a start. Please use the mic.
So my name is Aaron Ciechanover. I'm a physician by training. I graduated the Hebrew University of Jerusalem, then became a military physician because in Israel there is a mandatory military service, a combat physician, and then I did partial training in surgery, and then I decided that I don't want to practice medicine anymore, and I went to do a PhD with Avram Hershko. And during the PhD, we discovered the ubiquitin system, which is an intracellular proteolytic system. It degrades protein within cells, unlike proteins that are degraded like in the stomach or in the intestine outside of the cell. And this is mostly a quality control system that degrades denatured, mutated, and otherwise non-functional proteins. Finishing my PhD, I went to MIT for several years and did a postdoc with Harvey Lodish, where he was generous enough to let me work also on the ubiquitin system, so I continued my own studies on the ubiquitin system, and after a few years at MIT, I returned back to Israel, and I've been a faculty member in the Faculty of Medicine of the Technion, which is our Institute of Technology, ever since. Since we discovered the system in the late 70s, I'm very proud that we used what we call old good biochemistry. Lots of columns to purify proteins, to characterize them. And there was no molecular biology at the time. There was no cloning at the time. The human genome was not known. It was very, I wouldn't say primitive, but in retrospect, looking at it, I don't think that the ubiquitin system that contains close to 2,000 proteins, it's about almost 8% of the human genome, could have been discovered not using biochemistry. With genetics, you can discover one protein by screening for mutation, but you can never discover a cascade. It's a very complicated cascade in order to degrade a protein inside the cell, because proteases are kind of destructive, and you cannot let them just float around. So you need first to signal the protein for degradation, so it will be recognized by an otherwise cryptic protease. So there is no way but to use classical biochemistry in order to purify it. But again, during the years, I have undergone myself, my laboratory, many revolutions. The first was the revolution of molecular biology. We are cloning, we are mutating, Then was the revolution of using genetic manipulation of organisms, starting with Drosophila. Now we're working on models in mice, and so on and so forth. So things are developing very quickly in biology, and you have to adapt. And even the classical biochemistry has been now replaced because we are expressing all our proteins using molecular biological technology. So we have undergone major revolutions up to this point. But I think that my luck is that I'm commanding the two worlds, the modern world and the old one, so we can flip over whenever we need to use different methodologies. Now, the system is a very important system. There are basically, I don't know of any major cellular process that does not use the ubiquitin system. And many drugs have been developed based on it, mostly for cancer, but now people are moving, the pharma companies are all inside it, mostly for cancer, for blood, for leukemias. And now the world is moving to neurodegenerative diseases. Let it be Alzheimer, Parkinson, Huntington, poly-Q expanded diseases, ALS, and so on and so forth, because these are all diseases of quality control. You might think of cancer, you have oncoproteins, proteins that cause cancer, that normally should have been removed but are not removed. Again, a malfunction of the ubiquitin system. In neurodegeneration, you might think of aggregated proteins that are disrupting the brain function or precipitating in the brain. Normally they should be removed, but unfortunately they are not being removed, and so on and so forth. So there are many diseases that we thought initially independently on them, but they can be rather gathered under a common umbrella of quality, protein quality control, PQC type of diseases, and the ubiquitin system is handling quality control in the cell, and hence misproper or improper function of the system leads to diseases, and the pharma company are walking in and developing drugs. So that's in a nutshell.
Thanks very much. Didier, what about your field of research?
Well, my field of research is astrophysics, so it's part of the physics, and it's it's a field where essentially we have to build anything we need, because you cannot really buy anything on the market here. Well, I happen to experience, using the very edge of technology to do an equipment for the discovery of the first planet. What we had as a challenge at that time is to develop a machine that would have the capability to make a specific measurements at an accuracy at that time that was never met. We're talking about building a spectra for stars and then computing from the spectra the actual speed or the radial velocity of the star. So the trick at that time, it was in the 90s, that we have been using is a series of new components that started to show up on the market, but were never really combined together and were never really considered for astronomy. One of them is optical fibers. It's something we all know and something we all use. There is not a single computer communication which is not done with this right now. Well, in the 90s, it was already kind of there, but they were not really specifically used for astronomy. The reason why is they were designed for working in the infrared or the short infrared, and in astronomy, the equipment we had were working in the visible light. And a new kind of fiber was available at that time. They were never, ever properly being used for astronomy. So we had to develop with the whole team a way to have this fiber as part of the equipment. And that was critical because this fiber enabled us to reach a very stable entrance injection of the light into the machine. Then we built a new kind of spectrograph that was based on the old-fashioned design, but with a new component. But the real key here is on the market, something happened as well on the computer side. We had in the 90s a small PC. They were quite widespread at that time, but they cannot do proper scientific job. They were very, very limited. They had far less power than the cell phone we have right now. And you cannot really use it as an equipment to crunch number. To do that, you need big frame machine, and it means going back to the lab, getting access to big machine, using tapes. I'm still young enough not to having used, punching a card, but it was not very far from that time. No, what happened is there were a company called the Cray company. They used to build this massive supercomputer at that time. And they were based on a new architecture, which is 32 bits, which is something common these days. And part of the people of this company left the company and decided they could propose, instead of the big machine, also a smaller box with using this chip, the 32 Cray. This is known as the Spark system, Spark architecture. And in the 90s, they show up with a new machine, a new series called the Sun microsystem. And that was a revolution for astronomers. It was really literally a revolution because you could have in your lab, next to your equipment, a machine that could do about the same that you would need for big equipment. This, combined with the hardware, combined with the optical fiber, was the breakthrough. Because by putting that together, I mean, we were able to produce a machine that reached the requirement in terms of precision, also treatment of the data. To give an idea, I mean, in these days, when you get a measurement of a star, to compute at the level we needed, that was quite sophisticated, the speed of the star, it was 1 hour of computing time. So when you have a couple of measurements during the night, if you want to make sure you compute the data before you start the next night, I mean, you need kind of a significant machinery to do that. And that was one of the use of technology. And I can bring plenty of other use on big frames, but that's one of the very specific use that I directly was part of it and benefited from the output.
Thank you very much. We'll touch on this topic again a bit later. Now to Konstantin. We've heard that methods are enablers in some way, one way or the other, revolutionary methods. And if I'm not mistaken, one of your first revolutionary tool, an enabler, was a piece of Scotch tape. Is that right?
Well, I guess it's just my teacher and colleague and friend Andrew Geim just gave a talk in this audience, I guess, right? So probably I suspect that he did talk both about the frog experiments and the Scotch tape experiment. So but then when you say it in the just putting it in the past, unfortunately or fortunately, this trick is still quite active in our lab and actually in vast majority labs across the globe who work on graphene and other two-dimensional materials. So I mean we can come back to this later. So my background is condensed matter physics. So I mainly studied mesoscopic phenomena in transport, in magnetization, in superconducting systems. Some time ago, we started to work with carbon materials, so graphene with the Scotch tape. Indeed, it was quite a change of topic for us. But then at the same time, we used the same technology, nanotechnology, to produce other interesting mesoscopic structures. So it's not only for electronic, to study electronic properties. I guess I'm not sure if Andre mentioned his frog story, but I'm sure that he mentioned the Gecko tape story, and then so Gecko tape is a product of the nanotechnology and the quite extensive use of the nanotechnological facilities as well. And these days, it's called so-called dry adhesives is quite a big direction for many chemical companies in the world. And so, but generally, as Kapitsa said, so Kapitsa is the guy who worked with Rutherford and then was stolen by Stalin into Soviet Union. He got his prize for the superfluid helium, and so he said one order of magnitudes in temperature, a new effect. So what we do in our labs, we just try to interrogate nature, and we try to put as harsh conditions as possible, as harsh interrogation techniques as possible. I'm sure we're going to be one day prosecuted by the Den Haag. course for this. What do we do with nature? But we just try to put it into lower temperature or higher temperature or high magnetic field. So my second attempt to get PhD, which also failed, was in the high magnetic field lab in Holland, in Nijmegen, and so generally, I guess we just need to figure out how do we make those interrogation more and more cruel. But in this process, the main tool is still your brain, I guess. That's and unfortunately, it's the tool which we still don't know how it works. So we just have to invent our own tricks how to make it working. We've been talking about the hardware tools quite a lot, and indeed, as I said, so new magnets, superconducting or resistive. We can go to lower temperatures, and commercially we can really lots of cryostats, types of cryostats are available now. But then there are also interesting tools in theory as well, right? When we talk about the description of our say something close to me in condensed matter physics, we describe metals or semiconductors. We used to describe it in terms of the electronic band theory, the valence band, the conductance band, the gap, the wave function of the electron. So the wave function, it doesn't exist, right? It's our tool. So we just use it as a tool, and maybe sometime later we'll figure out that some other description, some other language would be more useful. So these days we switch to more topological description of material. So it's a new tool as well.
Thanks very much for these first insights, and I'd like to bring up a first question from the online audience already, which fits very nicely in this first round of questions. What role can quantum technology play in the advancement of biology, medicine, physics, astrophysics, material sciences? Because you brought up theoretical tools in theory, as you call them, that can advance scientific breakthroughs. Quantum technology is somewhere at the interplay between hardware solutions and theoretical solutions. Maybe you could go on right away.
Well, okay, so I'm not really working on quantum computing myself. I do follow some of those experiments, and I'm sure that there might be some people in the audience who are much better than I am in the field. But I guess there are many different applications for the quantum computers these days, but one which, if we're talking about tooling, so one would be the quantum simulators, and usually, so if you get two-level system, so you can always try to simulate the Ising model, and you can arrange your systems, whether it's Rydberg atoms or cold or ions or Josephson junctions. You can arrange them with this specific interaction to mimic a particular geometry, square lattice, hexagonal lattice, whatever, and then many problems can be mapped in terms of equations on this Ising model, and then you can vary parameter, and you can get your quantum simulator. So I guess these days there have been some discoveries of the new quantum phases obtained through this method. It's still unclear whether it corresponds to any anything in reality outside of that quantum system. But still, it's quite interesting because it just shows you a variety of the quantum phases possible, macroscopic quantum phases with the macroscopic order parameters. So then it might be it might serve as a guide towards what can we search for in terms of the new quantum states in the real materials.
Thanks very much. Aaron, if you had all the money in the world, what would be the tool you'd like to have, like to build?
Well, if I have to think back on the major technology that really revolutionized biology, is of course the discovery of the double helix. There is no doubt about it. I don't think that even Jim Watson and Francis Crick did understand at that time the full extent of the importance of their discovery, though they hinted it in a beautiful one-page Nature paper. So on the DNA basically lies the entire foundation of modern biology, and it ramifies to all directions. It was the beginning of decoding of the world of living organisms, from viruses to bacteria to plants to animals of all kingdoms and so on, understanding the code that make us what we are. It was the foundation to understanding mutations and diseases. It was the foundation of modern technologies like CRISPR-Cas, where we are now able to correct genes and to edit genes. We'll talk about bioethical issues that are related to it. It's not that simple. Modern drugs, understanding diseases, cancer, and so on and so forth. So, and now it ushers in this technology, I mean, in big, a new era in medicine, which is probably the biggest revolution that medicine has undergone in its entire history, which we call the revolution of personalized medicine. So until now, until I would say maybe 20 years ago or even less, medicine was kind of a very primitive profession. It was a profession of one size fits all. If you had a tumor, we didn't understand the reason for the tumor, we couldn't analyze it, so we bombarded it with chemotherapy, with radiotherapy, with mutilated surgery. I myself, as a surgeon in the operating theater, witnessed it. And now we are able to look at different diseases, including cancer, in a very sharp pair of spectacles. We are able to understand exactly the mutation that leads to it, or the cascade of mutations. And we are able to treat these mutations with very sharp tools. So if you have a mutation, now you can model the protein. We don't need even structural biology anymore. The classical structural biology, we can use computers now. We can use AlphaFold and Meta, all this software that can predict at very low resolution structure of proteins, and then computational chemistry can fit in drugs, small molecules, in order to modulate the activity of those mutated proteins. So think about what this revolution, what this discovery of the double helix brought to us. And I think that whatever we are doing in the lab, whether we are generating a novel species of mice, of a mouse that have a disease, whether we are treating the mouse, whatever we are doing in the lab is relying all on this, and the revolution is keep on running, bringing more problems of ethics. Of course, we can devote some time to discuss it, but I think that modern biology relies at large on on a huge tree that stems from this small discovery made in Cambridge in the mid-50s.
Thanks very much. Before we go on, I have a number of requests from our online participants that we are encouraged to speak up. Apparently the volume online is a bit low. Maybe our technicians could take care of that. Thank you very much. Didier, the James Webb Telescope, is this something you use? Is this a revolutionary new tool?
Well, I think any tool that will be able to see something that was not visible before is a revolutionary tool. So it's a long history. It started with the Galileo telescope, 400 years ago. Yes, it's a new tool. Yes, it's a revolutionary tool. It's the first time there is such an equipment that we're using. It's not the only one. There are massive, big telescopes under constructions, also on the ground. We are developing the community, the biggest ever radio telescope in the world called SKA. So the number of this gigantic equipment always existed and will always exist. What I would like to do here is maybe to put that into perspective. I think to build such an equipment, it takes really a long time. There is about 30 years until the time when you think through this, when you assemble the technology. It's so long that usually you know that there is some piece of the technology you don't have. SKA is one example. SKA doesn't have the computer resource to just treat the data. It's not a problem because extrapolating about the power of the computer in 10 years, they will be in 10 years. So I say physics in the general and scientific in the general, when you have to do something of that scale, you have this time scale. I quite got asked about why would you do that? Should we not spend the money to do something else? On that, practically, when you see how much money it costs and how long you need to do it, it's very little. It's almost nothing, I think, what you spend in terms of development here. And you can compare this kind of big equipment. ISS is one of them, and you can do a long series of this magic special equipment. They're a bit like in Europe Middle Age, the cathedral. I think they are the pinnacle of the technologies, and they bring people together. They bring people together with a vision. They really want to do something which is new. 1,000 years ago, it was inspired by God. Right now, it's driven by this kind of strong impulse to understand and to see through, to see back in time, to see the beginning of the universe in the case of the James Webb Space Telescope. So there is a long series of such equipment. Another one, completely different, will be at the CERN when they're trying to understand the very intimate interior of the matter, also with similar kind of scale and similar time scale of development, and also some kind of global effort. I think I would like here to emphasize something that was mentioned many times during this meeting. This is not just not few people in the lab and playing, having fun here we're talking about. This is thousands of scientists that doesn't share the same language, culture, and country of birth, but they come together. They come together because they need to be together to do that. And I would like to mention something about what is the best equipment you may think about and related to that. I think there is something that to me is just amazing, and you mentioned it actually. This is the brain, and what makes us so special is these capabilities when you compare to the other alive animals or entities on Earth. Is this ability of the brain and to think through, to imagine, and to do something that we never thought would be possible. So if I would push what is the best machine, I think I would certainly look at the brain. And one way to make it, which would be very simple to improve, is I think we use little the many brain we have on this Earth planet, and practically here. When you look at how many countries, how many people have really access to high educations to push the brain, it's a small fraction of what we could do. So if I would try to promote science, the best equipment, I would try to generalize access to educations all over the world for any continents. And until I have the same amount of people that is producing math coming from Africa than we do have from other continents, I think I would feel that we have failed here. So I think this is really what we have to promote. Anything we do on the top of that is just the creation of the brain. And that's unbelievable when you think that 10,000 years ago, we were, essentially fighting for food and for survival. And it's amazing right now we are 8 billion of inhabitants on this planet, and we kind of fix the problem of food. And it just seems so magical. So this would be my kind of take on this.
Yeah, can I check on you? So you just mentioned that you started those projects like SKA and web without even having the right tools, just hoping, projecting that it will be developed, in particular in computing as well. And I know that astronomy relies quite a lot on the citizen projects.
Yeah.
Some of them, I participated in some. So, and in many of those citizen projects, you don't even need to use the brain, just use the few neural network layers like in your retina, and that's it. Do you see those students and citizens like me as the tool as well, as consumables? And
That would be an amazing already citizen if you want to look at this. Yeah, definitely. You know, it's one of these many opportunities we have here, clearly. What is nice with technology is within a kind of horizon of 5 years, we can kind of guess a bit what to expect. What is impossible to guess is about, fundamental research. It's impossible to guess what is going to happen in 5 years because this is the unknown unknown. And while technology is something you build on something you know, so you're trying to push it until some extent until you reach some goal. So some of the development are being made with a perfectly clear idea in mind what technology is doing right now, and the idea in the labs right now how is going to be brought together to reach a new tool, certain new equipment, which is a major difference between predicting the technology and predicting the new knowledge, because in that sense, new knowledge by definition is something you don't know.
Absolutely. I'd like to encourage you again to speak more directly into the microphone for our online audience. Carol from somewhere in the world asked a question that is closely related to what you've just discussed. In your opinion, does technological advancement drive innovation, or do we need innovative ideas to develop revolutionary technologies?
I mean, first of all, we're talking about we're talking about science here. So in terms of innovation, probably yes, but science is different from technology and from innovation. As Didier said, it's the unknown unknown. So we basically what we do globally, and we just apply some pressure on a bubble, and we just hope that it will be, and we expand gradually the level of our knowledge. And then at a certain moment, this pressure bursts and bursts this bubble, and we expand into the new territory, and the new knowledge, the new concept appears, and then that's the scientific breakthrough, and it just defines a new frontier, and then a new knowledge pressure has to be applied. So the question is whether this burst happens because of the new tools. Well, I would say, of course, it has relevance, but usually, though this burst and the new ideas is more a product of the collaborative research and deep thinking rather than one particular tool. So the tool produces data, and it keeps producing, and it's difficult to say which particular piece of data triggered this. So I would say that it's necessary, but it's not the crucial moment.
That is an interesting statement. Aaron, is this along your line of thinking?
I think that in biology, the answer is dual. Yes, innovation drives technology, and technology drives innovation. Because once you are this, I can bring you one example, again, from the world of DNA. Initially, people learned about the double helix. Then they wanted to understand how the information is embedded in the DNA. So they looked into sequencing. So they invented the way like Wally Gilbert and others invented the sequencing of DNA. Then other technologies came. Think about microscopy. We used to use light microscopy. Then came electron microscopy. And recently we wanted to understand the molecules, how they act in the live cells. So super resolution microscopy came in. So questions are driving technology, and the technology brings no questions. So I think it's kind of a cogwheel that is working in biology for sure. And the DNA, people say, okay, the DNA is making RNA, so let's go to RNA. So they invented methods in order to sequence RNA, but then came a more difficult technology, which is how to sequence proteins, because DNA and RNA are made basically of the same basis. But proteins are made of completely different components, of amino acids. So then came proteomics, and so on and so forth, and post-translational modifications. So you see that, one technology drives another question, the other question needs a new technology in order to resolve it, and so on and so forth. And I can bring you numerous other examples from the world of biology, how this is a kind of a cogwheel.
So I like to give the physics perspective of this.
Please do.
Astrophysics. Okay, so and I all agree with what has been said. From a physics point of view, there is something that we can absolutely say is technology is entirely, I said entirely driven by innovation. So this is where technologies sit. Well, the opposite is not true, actually. Innovation is not entirely driven by technology. It is in most of the case, but there are plenty of good examples. And my best example is Einstein sitting at his desk, thinking about beauty of time and space, and something doesn't fit exactly together into the physics which is known in these days. And he's coming with this beautiful theory, which is certainly one of the most beautiful theory that we have in physics, which is general relativity. Well, this is not driven at all by technology, except the technology of the pen and writing of the paper. But instead that, it's completely come out of his brain and with interacting with the rational thinking of the physics. But I can tell you this new invention, this idea, this physics pattern of the universe is completely used by the technology of your cell phone. It's called GPS. So it's one of the bad examples where it doesn't go always in both directions.
Right. So just pouring money into a system to develop new technologies by very skilled engineers will not do the job.
Well, the most famous, I think, sentence, I don't know who started, I think Gilles was one who said it, is you can spend all the money you want to try to increase a technology, and the one I like to use is the steam engine. You use the steam engine, it's great, but you're never going to fly to the moon.
Let's come back to the question of the role of the human scientist, the human brain. Artificial intelligence is back. We have seen several waves of machine learning and AI throughout the past, say, 50 years. And we can expect that AI is back for good right now. It's here to stay. And there are several tools available belonging to the field of generative AI. Do you think these tools can support you in hypothesis formulation and actually coming up with ideas you have not thought of before? To which degree do you use machine learning or these deep learning tools for data analysis? Let's start that way. Konstantin.
Right. So it's absolutely clear that AI and machine learning increased our ability to do research and produce new data dramatically. So the beautiful example is the protein folding by AlphaFold, and I would say unfortunate example is the automated translation, which basically, for me, it killed such a beautiful area of mathematics as linguistics. So, which personally I really regret a little bit. But okay, that's life. So that's how it works. So, but it is clearly an amazing tool. And I guess we really coming to the question which you and I should say that we use it in our work as well to do calculations on very complex structures which you won't be able to do, or you would need months and months on the most powerful supercomputer we have here in Singapore. So it's quite it is a beautiful tool, and if used properly, then it works and gives fantastic result. Now you've got your this beautiful question whether it can assist in hypothesis formulation and in hypothesis verification. That's it's still a big question for me. Say, I'm sure that in some cases where the number of possible hypotheses is limited, I think it will work. It will work really well. So, for example, if you are in medicine and you need to do the diagnosis, then you can probably formulate a hypothesis and then try to verify it and produce a series of tests required, which it needs to verify. Would it be able to recognize coronavirus back in 2010 through this? I don't know, because it is the production of the new knowledge. So for me, it's still an open question. However, say even a year ago, I would give a straight no to it. I'm so confident in the human brain that even though we don't understand it, but somehow magically it gives us the opportunity to produce discoveries and make the breakthroughs. I'm not so sure now that I would say it's not straight no. So it's no with some doubts that it probably can. There is some chance that it will be able to produce completely new hypotheses.
Aaron, Didier, do you concur with this conclusion?
Yeah, in medicine, AI is a very important tool, and I emphasize the word tool. Tools are developed by people. And so it's only a tool that helps us to advance knowledge. And in medicine, there are many ways that people are using now AI. We in the lab are using it for pathology. You know, pathology is still a profession of pathologists. They are using microscope in order to tell you whether there is cancer or whether there are processes. The advantage of AI is that we don't know what it's doing. We simply don't know. It gives us information about things that we have never thought of. You know, for example, you're looking at a cell. The pathologists look at some features, but the eye is limited, and the brain is limited. But the AI can look at zillion features at one time. So let's say that it can see a corrugated piece of a membrane that is like, and it's associated with a mutation. So it will point out to you that in this type of cancer, the mutation is associated with this corrugation. So now you can use a new biomarker in order to diagnose the disease. And if you follow the patient, it may give you a prognosis, a success of treatment, and so on and so forth. So the idea is that AI can see what we as people, our tools, cannot see. There is also a big movement of moving away from animal experiments in order to introduce new drugs to the market. There is a big public resistance to using animals, but on top of it, animals experiments are extremely slow. You can take, let's say that you need to screen 10,000 drugs. So how do you do it in animals? You're taking 10,000 mice and you inject the drug and you develop the cancer and you're following and you extract the tumor and you give it to the pathologist. It's an endless process. Therefore, development of a drug nowadays takes something between 5 to 10 years, costs billions of dollars, and the same for clinical trials. So what people are using now, they're using organs on chip. So you can make from stem cells a little liver or a little fatty tissue tiny, just a cluster of cells. And you can, with micro tubing, you can flow fluid via it, and then you can screen thousands of drugs at one time. And you can measure the output, what's going on. So you can measure toxicity, and you can do also side effects of potential drugs. And the only way to analyze it is by AI, because the output is huge in data. And again, the computer can combine all this data into a cohesive picture. And on the opposite side, you can develop drugs, not only monitoring toxicity. So you can take stem cells from patients, or you can take blood and convert it to stem cells and then go to any tissue you want, and you can develop personalized drugs. So again, you are doing these little organoids, and you are trying drugs that will fit the patient. And it's very easy to do because you are screening zillion of drugs in no time and almost with no, I wouldn't say with no money, but much, much, much cheaper. So AI is clearly going to accelerate in a big way and to lower the prices, to accelerate the development as far as time is concerned, to lower the prices dramatically, because an average drug now to bring to the market is something between 5 to 10 billion US, about.
Yeah, I think I would absolutely share. I think it's a tool. It's nothing else, nothing more. There's a bit of a buzz on this, but it's just a tool. Whether I'm using it, yes, every day. When I want to log in into my cell phone, it's just using AI anytime. It's a pattern recognition mechanism. Of course, we're using it. I'm using it a lot in the translator. It's a fantastic translating machine. It's a pattern recognition system. So it's a tool that is based on a pattern recognition, whatever pattern means. It means you have a model, you match it closer to your expectations, and then it's something that you put together. No, there's nothing brilliant, there's nothing brainy, there's pretty much stupid, by the way. It's just pattern recognitions. But that's how I think it brings very impressive result, because it do it very fast. And it's possible because of the computer power. The idea of AI is very old, actually. And it has evolved because there's a lot of different mathematics way to do that. But since now the computer is so powerful, this is the most brutal, stupid use of a computer, which is trying to match all the possibilities with a kind of an optimization way and different filter. Trying to reproduce what is going on in your brain. Well, I do think something else is going on in your brain, and we just touch the real pattern of what the brain is doing, the functioning of the brain. I don't think AI give us any clue about consciousness, and it would be nice, but I don't think it does. As a tool, it would certainly help me, help my research. If I have zillions of data, I want to clean it up and get like a filter approach or simplify the data to be able to extract something out of it. But I doubt AI will write something really new. He may certainly reproduce plenty of fake papers, and there's a lot of games on that, I'm pretty sure. You can play with art, you can try to simulate many different pictures together, recreating something. This is fine. It's a tool, and nothing else, nothing more, and there will be more tools in the future that people will invent with the new technology available.
I can actually say why I'm less pessimistic, or whatever, not pessimistic, but skeptical than Didier. So generally, they say that a smart guy is someone who can make an analogy between two events, and a genius can make analogy between two analogies, right? So you basically learn from something else and apply, and somewhere at a certain moment, even without your participation, your consciousness, your brain makes this comparison, and just and one event triggers another event. I don't know, you drank coffee and you saw some waves and interference in your mark, and you thought about just interference pattern somewhere else, or you even not interference. So you never actually know what triggered the discovery. So what's really puzzles me now in AI, the use of the pre-trained models. That's how something which was trained on completely different data sets being can be used for, I don't know, something trained on the pictures of cats and dogs can be used to describe the quantum states of the molecules. It's beyond my understanding, but somehow those pre-trained models been shown that we've been shown to work better, and that's almost magical for me. So so you're just making this analogy between two analogies.
You've touched on a hot topic, that of human creativity, stupidity on the other side. Let me take you up on that, because we have several online questions revolving around this topic, and Vijay put it very nicely. How can we ensure that the use of technology and revolutionary tools in scientific research do not lead to a loss of critical thinking and creativity? Loss of critical thinking through the use of advanced technology. Is this a danger?
I think it was always considered to be a danger, and then I mean, let's be honest, science is not something which we do only now. It's been active for 500 years, and I'm sure that every single generation thought, okay, now science is that. So Newton invented, Leibniz invented the calculus. So everything can be done with it, and then so electromagnetic waves and the four equations of Maxwell. Okay, now we really understand everything. I mean, I'm using the mathematical tools as the example of the tools, but you can say the same about many other tools, and yet it just every time it has been shown that science and nature is by far deeper and by far more interesting, and it's clear that the number of questions which we still have is huge. Frankly speaking, the more I study physics, the more I understand the vastness of this unknown territory. And we already mentioned several times that yes, the technology and tools, they create this global pressure, and they gradually expand this known territory. But this burst, this breakthrough, it happens somewhere in our brain, and only in our brain. So I don't think it will. So the answer is that probably I can imagine some scientists will be satisfied with the little petty work which they do, but the breakthroughs are going to happen, and they're going to happen in human minds.
I think the question is quite interesting because it shows that there is this fear of the technology, and pretty much it's not just now. It always happened. Anything new coming up, there's some people that are a bit afraid, and because they lost control, they don't understand what's going on, and because they don't control, they don't understand what's going on, they want this to be removed. And I think it's possibly the way we react to the unknown. Now, we know, you can bring a child and to any place, he would just catch up the language, he would catch up anything, and we can even keep doing that because the brain is very plastic. So there's no reason to be afraid of the technology, and if we do feel so, I think there is something about the understanding of what's going on that should be threatened. I would be much more afraid of governmental policy, restriction of travel, and restriction of thinking, expressing yourself, than really by the nature of the technology itself as preventing creativity here.
Not being afraid of technology also means that you need to be knowledgeable of this technology to understand the mechanisms of this technology. Aaron, is there anything you could recommend maybe our audience here, mainly young researchers, when it comes to new technologies and maybe being afraid to touch these seemingly complicated tools?
Yeah, we are clearly living in a different generation nowadays compared to my generation. I think that I'm the oldest among the panelists, and when I started my PhD, There was not even pocket calculators. Only towards the end of my PhD, I bought for myself the first pocket calculator that could tell me that 2 and 2 are 4 and the square root of 9 and sinus of a 30 degrees angle and so on and so forth. And now it's completely different. But I think that the passion of people and the drive of people has still remained. Now we are talking about a new AI program, the chat program that can compose for you anything you like. You know, you want to write a work in your university about the history of the covalent bond, just kick it in. In 2 minutes, you will get a beautiful piece of written work, a study, and so on and so forth. The question is whether it's innovative, and probably it's not innovative. It's more historical, it can compose things, and I think that also proud people, and I think that I am very proud, want to do it on their own. So at the end of the day, the real driven people would not like to rely on such tools. They would like to go to the library to collect the data, but then to think, okay, where the data are leading me? What can I do with them? How can I use them to innovate something, to invent something? And I think that this will be the limitation of the tool. I cannot prophesy that the tools will never evolve to become innovative, but I think that, you know, in our hearts, we are still driven. There is something about us. And there will always be these driven people that would like to do it on their own. And I wish that many of you will be like that rather than to push a button on your keyboard and get something that is ready to go without even reading it and submitting it to your instructor in the university, graduating without knowing even the alphabet, which looks to me like an oxymoron.
I think it actually brings this important thing about collaboration. So that's what I mean, that technology is important and tools are important. What is important is that you know how to use this tool. So every single tool got a limitation. So, and my first, when I started to do my first PhD, my supervisor always said, so you have to love and know the device which you use, because it's still a tool, it's still a device, and it has many limitations, many boundaries, so each technique got those. So you have to be, so even the best tools, they're not useless, but they're not as efficient if you don't understand how it works. You really need to know it, and it basically means that you have to master the technology, and unfortunately you don't have chance to master all possible technologies in the world. So what is important to have is to have, like what physicists do, is to have a mental model of your system and understand what do you want to study, and then know the possible techniques which would allow you to do it. And of course you are never expert in all of them, so you have to collaborate and work with someone else who can help you to interrogate your system from a different perspective.
Thank you. Didier, your recommendation.
My recommendation?
For the audience, for the students.
Well, I think they should not be afraid of the technology, but I don't think they are. And if they are in this audience, it's because they're very curious and they were very keen to learn. So I'm certainly not be worried about this. What is interesting is to consider themselves as maybe an advocate of technology and understanding how knowledge is critical for the society, how to translate the knowledge into a global scale to the society. And in this audience, we are the future generation of really of promoter or advocate or diplomat of science, and we all have a task. So I really hope that you take this task very seriously, because if you don't do it, who else is going to do it? So that would be my advice.
Wonderful. Now we've been addressing you, the audience, here in the lecture hall, and I'd like to invite you and encourage you to ask questions yourself. You see a few microphones lined up here in the hallway. Please line up, go to a microphone. I will call you if you have a question. And while we have the audience lining up to ask you?
Already aligned.
Already aligned? Okay, then why don't we start with this?
Hi,
Over there, please.
Hi, professors. Thank you very much for the discussion today. So my question is, as my friend used to say, the easy thing has already been done. So the next thing that needs to be done, like the revolutionary science, needs to come from the revolutionary tools. The problem is it leads to the concentration of resources. So yes, if we are in a very prestigious group, the resource is coming naturally. But what is the advice if you have for the people who are not as blessed for the resource that doesn't have the access to those tools that are required to make such revolutionary discovery?
I would say I completely disagree that all the easy things have been done already. So if you look at the history of discoveries, so many of the discoveries which to us now look completely obvious and just how it could be any other ways, they had very, very difficult time to be published and to convince other people. So I don't like this statement that all the easy things have been discovered. They look easy for us now, but at the time of discovery, they look, believe me, they look very, very, very controversial. So Klaus von Klitzing, he's not here, but so he discovered the quantum holofact. So something which is now, I don't know, it's being used in metrology widely, of course, and it's the very basic concept for any condensed matter student. He had terrible time trying to publish his work because no one would believe him. Even now, so today, it's something as basic, I don't know, for any condensed matter physicist as possible. But then, so that's the first statement. But I would say that the lack of tools make you more innovative, probably. So you can really try to trigger your imagination. So. I'm sorry to put my case forward here, but so we started to work on the Gecko tape and on graphene just a few months once we moved to a new lab in the UK in Manchester. So I entered into the lab. So there was nothing there. So there was a turbo pump and a desk and a chair. That's it. So just really completely empty, and believe me, we didn't need our first devices. Were literally produced by hands only with the stereo microscope. So I don't think. I'm not sure. Okay, I wanted to ask Didier if he says a direct linear proportionality, or I don't know, a square or square root dependence between the price of the tool and the number of discoveries you can make per the unit of time. But I'm not sure if there is such a. So can you plug the log plot?
I think this is a very interesting question because there's really two approaches here. We call that large science, when there's usually very expensive equipment that usually is assembled and thought carefully, where there's many people building up. And sometimes this equipment are making something new and they're producing what they expect. But there's also a complete other part of the science, which is exactly what you described, which I would qualify also from the first discovery of exoplanet. At that time, I think the funding was almost none. And we have to do everything with a shoestrings. And there's plenty of examples like that because we're too much ahead. And we're too much ahead of the time, and nobody believed that because it's not yet as part of the streamline of the science. So if you start science and feel that you are not in the right place, don't have the right finding, I think you should ask yourself what exactly you're looking at here. And maybe you're not really looking at complete discovery science. You may be more more interested about applying elements and developing tools and stuff, which may be a different aspect of it. But the true discovery comes from nowhere, at a completely unexpected time, and it should not depend on the money. Money helps. It's always good to have money, but the long list of examples can be produced demonstrating that certainly it was not the trigger of discovery. And when we announced the discovery of the planet, and we had a big problem, and in a way people were reacting, oh, how come such a big discovery has been made by only 2 people? You see this how it works. So sometimes you expect big stuff. We're talking about James Webb Space Telescope. It's a great tool, thousands and thousands of people. Is this equipment going to do a discovery that will lead to a Nobel Prize? Not sure. So maybe it will come from next door.
Very discouraging to the people who run the Webb telescopes.
Yeah, well, and there may be some surprise there, but I think essentially the investment you need to do when you have big money can be predicted to some extent, because that's how you get the money. If you really want to go into unknown territory, the money will just shrink. And the more it's unknown, the more it will shrink.
Thank you very much for this answer. You see, with your questions, you hit a spot here. Absolutely. Thank you for that. Please, we have another question to be asked.
Hi, Professor. I think general relativity is mentioned just in the talk, and it's a typical theoretical physics. And my question is, what do you think about the relationship between theory and experiment? And I think often they have disagreement, like, for example, like some experimental physicists doesn't think, I think string theory is not physics, but just mathematics, because no experiment supported. So I want to ask, what's your viewpoint about this?
Well, I think there is something that would qualify for anything which we call science. It's not only is physics, it is what we call fact-driven. That's it. So you can come up with any beautiful theory of the world. If there is no any fact that drives the theory and brings the theory, it means nothing. So it's really fact-driven. It's not driven by a book or by somebody, promoting a kind of mental picture of it. It's driven because there are facts. And if you think about the Einstein theory, at some point Einstein was very eager, predicting some of the fact that could be checked, and then that made the theory even stronger. And there's still people challenging a theory these days by observing deeper and deeper and better and better. And it qualifies for any field of the science. It's fact-driven. So it's irrelevant, this debate about theory of observation, because the fact is the driving force of science.
So you see, you asked the wrong people. So you've got the bunch of experimentalists who just who are going to screw now badly. So I completely agree. Yeah, if your theory doesn't, so of course theorists would say, just if your experiment doesn't fit into my theory, go and measure better. But if we say just if your theory doesn't fit with the facts, probably it's not beautiful enough. So
yeah, never repeat a successful experiment. Absolutely right, Aaron.
Well, I think I cannot compare biology to physics. I think that the physicists are kind of aiming more at the unifying theory. In biology, there is no unifying theory, so biology is much more eclectic. Some people work on viruses, some people work on the brain. There is nothing between the brain and viruses, except for basic elements like DNA and RNA. But otherwise, the mode of action are completely different. Some work on plants, some work on symbiosis. It's completely eclectic. And at the end of the day, biology is so complicated that you need at the end to experiment it in a wet way. If you are developing a drug, at the end the target is the human being. And I don't know that there are not two human beings that walk on the face of the planet that are identical, not even identical twins, because they are different epigenetically, because they acquire different methylation of their RNA, of the DNA, and so on and so forth. So the response of one person is not the response of other. Maybe one day will come and we'll have so much information that we'll be able to predict a priori that with your genetic repertoire, the drug will do that and that. Until then, we need to experiment it and just wait for the result. So I think that in biology, there is much less place for a theory, and at the end, you have to go to the sink to turn on the faucet, wetten your hands, and do it wet-wise.
Thank you very much.
Okay, thank you
I suggest we continue on this side and then over there.
Thank you.
With your many years of experience in science, I was mostly wondering how you feel about tools that allow collaboration, which we've recently seen with Zoom and online meetings, online conferences. Do you think this will change the way we do science or the way we collaborate going into the future? Or maybe with your 40, 50, 60 years of experience, how would you say that these tools have changed the way you're doing science from when you started to how you're doing it now?
Thank you.
Oh, I guess.
Well, I can start easily. I mean, the web was created by the physicists that needed to exchange data, and we've been the first to use the email way before it became into the public. So science is about communications because you need to exchange, to challenge your ideas, and you need to communicate. So anything that enhances the communication is good. It does not magical because it doesn't make the communication, but it enables the communication. So great, I think I'm a great user of Zoom communication, any kind of tool like that.
Me as well, but I still way prefer the in-person meeting. On the Blackboard, I don't know what's the magic the Blackboard got, but it's so much, you just managed to go through so many more models within shorter period of time. So if I would definitely always prefer in person, and I mean, come on, you cannot you can just call in person immediate on Zoom doesn't have exactly the same connotation as calling person immediate in real life, right?
Does this answer your question?
Yes, yes.
Thank you very much.
It's also, has it changed the way you're doing science, or do you think it will change the way we're doing science?
Only we knew how the science is done. So the science is done in your brain, and then what triggers it, we have no idea. I mean, at least I have no idea.
Well, we wouldn't have such a discussion on Zoom, I guess. Okay, thanks very much. Next question over there.
Yes, hello,
please.
thank you. So you've talked about, let's say, the capacity of the brain and how unique the brain is in generating ideas and drawing analogies. So one thing that puzzles me is, let's say, the amount of information that we can process. I mean, I think in let's say, publishing in science, I think in the, let's say, since the 2000s, the number of papers that has been published is higher than all the papers that were published until the year 2000. So I feel that, let's say, I mean, we still generate ideas in the brain of a single human being or maybe in a collaboration, but do you see, let's say, the amount of information that we generate? I mean, let's say there's, of course, a benefit in it, but I guess it's also a challenge, let's say, in the way we do science, that as a single scientist, we can only process a limited amount of information to generate new ideas. So I see it, personally, I see it as a challenge because I feel bombarded by information all the time. So how do you see it on a personal level, and maybe does that require new tools that kind of pre-filter information, or does it change the way we do science on the long run?
I'm not an information scientist. I'm sure someone with the right background will be able to give you more proper answer. But here is my amateur approach to it. So I would say that probably when our brain fills up, we just die. So that's why we live so short. So things like trees, they don't need to bother to process information and fill up their brain, that they can live for thousands of years. So, but then you're right. So the amount of information gets larger and larger and larger, and that's where the physics approach help, where we create a model where we just digest and dissect the system into some basic parts, and then we just and then the model is basically archiving all this all those data into the digestible form. And then once the model is created, there is even better way to archive this information. We create a new language. So I mean, if you I don't know. So just we talk about sorry to get into my territories. If you go into the condensed matter physics, we talk about electrons, you talk about holes, even though none exist. So it's some excitations in there. So in the Fermi sea or whatever. So, but then we just learned how to we create a new language, a new operator, a new mathematics, and then it's becoming way more compact. And if you look how we manage to compact our data over time, so the growth of knowledge is not exponential, so it's actually quite slow in that respect.
Thanks very much. I suggest, in light of time, we go straight to the next question over there, please.
Hello. So I have a question regarding interdisciplinary research and the creation of new fields. Like, for example, these days, if you do research, start doing research, you have a lot of data over the internet and you have a lot of fields, and every day there's a new paper on every different topic. And so I wanted to ask you because these days we all have, not all, but most of the people have primitive mindsets, and it's very difficult to create a new field. To create a new field is very difficult. What tools do we need? And for example, let's say cancer. It's a very big topic. Every day we have a new paper on cancer. But what would it take to answer that question, to solve the puzzle? Will it take a new tool, a new field? What would it take to answer that question? And the main question is the creation of a new field from current disciplines.
Oh,
yeah, sorry.
I don't know why you have to generate a new field. You talked about cancer, didn't you? So I can bring you a zillion of questions about cancer that are still not answered yet. For example, why cancer of the skin is so easy to treat, while pancreatic cancer is so resistant to every chemotherapy. The same for ovarian cancer. What's going on in these different tumors that are making them different from other tumors? So there are so many open questions, even within the big definition of a disease, that are still completely available for investigation. Why do you need to open a new field?
So you mean that creating a new field is not sufficient to answer all
Yeah,
the questions?
the question is, what is a new field? You can invent a new chemotherapy, a new class of drugs, I can bring you many examples of new drugs that are entering. Cancer therapy, for example, moved from chemotherapy now to biologics. We are using antibodies which are very specific, like Herceptin and others that are targeting very specific molecules and have much less side effects than the chemotherapy that is non-specific. So here you have a new field within an already known field. So I think that, there are always new questions that are coming up. You don't have to bother yourself with something, well beyond your ability or something that you have to prophesize. I think that there are so many open questions. Think about Alzheimer's disease. Think about all the neurodegenerative diseases. What's going on? We have no clue what's going on, but even the slightest clue. We don't understand memory. Think about these diseases. You are losing memory, right? So it's already known disease, Alzheimer's disease, and people are losing memory. What is a memory? That I'll tell you your mother. So all of a sudden pictures are coming into your mind. Smells of food, of ceremonies at home. Where do they come from and why they get lost? What happens? What is the connection between aggregated proteins and memory? I mean, there are so many open questions in already, I wouldn't say known field, in known entities that we are starting only now to unravel, that I wouldn't be bothered at all. I would not be bothered at all.
Yeah, I would like to push in this direction even a bit further. I think science is very organic in the way it's being built. And look at the double helix of the DNA. When Crick and Watson, as a physicist, playing with an equipment of physics in the physics lab, well, in a way you can say they created a new field, but actually this is just what they found was so interesting that it attracted a lot of scientists, and things will be very organic. So there is a little bit maybe of a point behind your questions. Is not about the science, it's about the curriculum that drives the science, because it happens that the way we organize institutions and also the funding agencies is organized into field. And that's maybe what you are, alluding on here, practically. And I think this is a mistake of the institutions that date from maybe the Victorian age, when we start to define fields in science. I think the science is not anymore so easily identified as field. And it's up to the institutions to adapt to these situations and to enable, I think, a culture that would be much more agile in the way it is being done. And that's why you see popping up in different places what we call cross-disciplinary centers or joint activities to try to remedy to this lack of creativity as part of the curriculum, the way it's being done. But setting up a curriculum that would be more open is complicated by design, because then how do you assess it, and how do you deal with a funding agency when you start doing something which is in between different kind of disciplines, and how do you assess that? So it's certainly not a scientific, element, because scientists can move from one element to another one and doesn't care about the field. It's how the society and how the practical structure of the governance of the science is going to reflect on that. That is really what you're talking about, unfortunately. And that is what us as a scientist are battling against quite often. Unfortunately, we're quite too much sometimes fighting wars because people, they don't understand how organic is science.
I wanted to echo Didier, so just 10 seconds. So you are educated as a biologist, chemist, physicist. The moment you graduate from your university, you shouldn't be allowed to call yourself a physicist or biologist. You are a scientist. You cannot just put the shores and then say, okay, I'm using this knowledge and I'm working within this field. You are a scientist, and science is global. Is the same physical laws which are operational in biology or in astrophysics. But again. It is true that we are educated as within a particular field, and maybe so now we try to mix and match different disciplines together. And there are universities like this which don't teach, say, physics. They teach quantum, which is mathematics plus physics, or they teach material science, which is physics plus chemistry, and so on.
That was longer than 10 seconds. Thank you for this answer. Quick question, quick answer.
I try to be quick. Thanks a lot for a very interesting discussion. My question is, recently FDA just pushed forward the idea of some medicine may not need any more testing. So this is an example of how science policy can affect how we do science. Can you comment more on this, and what could be the challenges? What could be the challenges, and also whether it's one of the ways to drive forward the innovation of new technology, new tools? Yeah. Thank you.
What was the question? I didn't get it. Can you repeat it?
Please rephrase your question. You had two questions in there.
Yeah.
So the first question is basically it's just one question. How do you comment on science policy? Something like for example, FDA just pushed forward the idea of some medicine may not need animal testing. For example, in biotechnology, we use a lot of animal testing, like animal models. But this kind of policy, do you think is really, in the future, could change how we do science in general? We need to think about alternative methods, new technology, new way of proving the hypothesis. So, yeah, I just want to hear what you think about this changing in the future.
Can you get rid of the animal testing altogether, and what
I cannot. Yeah, okay. The FDA just 2 weeks ago issued an instructions or a new law. It depends on how you look at it. That in the future they will approve drugs without animal experiments. But this is a little bit too far. So they opened the door to this one, and I mentioned in my brief talk, in one of the brief statement that I gave here, that we are moving now to organoids, to tissues on chips, which might replace the animals. What we'll need is several drugs that are using the two methods, and we need to correlate, because animal experiments are okay, they are not ultimate. You know, most of the experiment, you know, how many mice were cured from cancer and how many drugs are failing in phase 3 clinical trials. I mean, it's easy to cure a mouse. It's very difficult to cure a human being. And most of the experiments fail at the human clinical trials at phase 3. But animals are still a whole organism. They have heart and blood and lungs, so you can monitor a lot of parameters. But people are moving from it, and for the new drugs that are coming, people will have to compare the two and to convince the FDA that these are parallel or even the organoids have an advantage over the animal, and then gradually, hopefully, the animals will phase out. More importantly, we are talking about clinical trials in human beings because I'll be able to take tissue from human, just to take a blood, and to make different tissues out of it, to convert it to stem cells, and then to take the stem cells and to convert them into the different, and I will be able to circumvent even clinical trials. This is a little bit of a far-fetched dream, but that might also happen. So it will save, again, a lot of time, a lot of money, and a lot of suffering, because there is an ethical issue with clinical trials. Because in clinical trials, you always have two groups. One group that enjoys the new drug, if it's successful, and one group that gets the old one and might suffer the disadvantages of the old drug. So there are also ethical issues in clinical trials. So I think that the world is moving for them, and I think that the step of the FDA was crucial in that respect.
Thank you.
Thank you very much. Thank you very much all for asking such wonderful questions. And time is up, I have to say. Dear friends, we only just started a fantastic discussion that led us from classic tools to far-fetched dreams. I thank the panelists very much for sharing their thoughts. I very much enjoyed your comments and contributions to this GIS panel, and I thank the audience, all of you, for attending and staying true on the path of science. Thank you very much.
Panelists for the insightful comments and discussion. We have come to the end of the afternoon session. For those who have signed up for this afternoon's small group discussions, these will start at 5 p.m. So there are several sessions that we held at the think tank rooms on level 3. So the first one for Prof. Brian Kabilka, it will be at think tank 19. For Prof. involved in the small groups, we will be starting, we'll be having the young scientist presentations. Yep, so while we thank you. So today we have two young scientist presenters, Sauradip Majumdar and Una Trivanovic. And we would like to invite Dr. Yen Ning, the Dean's Chair Associate Professor of the National University of Singapore, to chair the session. Dr. Yen, please.
All right. Good evening, everyone. I'm Ning. Now I'm working in National University of Singapore in the Chemical Engineering Department. But before I come to NUS, I spent 4 fabulous years in Switzerland. So that gives me the great pleasure to introduce 2 presenters, young scientists that are going to give the presentation. Both of them are coming from Switzerland. And that reminds me of my wonderful student years. The first presenter is Sauradip Majumdar. He's a doctoral student at the Swiss Federal Institute of Technology, Lausanne. His research interests include computational chemical engineering, molecular simulations, machine learning, and carbon capture. Allow me now to hand the time over to Sauradip. Yes, please.
Am I audible? Okay, so hello everyone. Very good afternoon, evening, morning to whichever part of the world you are all joining from. Today I'm very happy to be here and share a little bit about my PhD research, which is titled Finding Promising Nanomaterials for Energy Applications Through a Diversity-Driven Search. Okay, now many of you might have come across similar images or news articles in the last few years. And in this context, I would like to share a personal story. This is probably last summer of 2022, when I was talking to my family in India, and we were discussing about temperatures in Switzerland. And I was telling them that today the temperature is around 38 to 40 degrees Celsius. And my parents thought that I was joking. Because typically, when you speak of Switzerland, the images which come off is something like this, a white area, glaciers, mountains, snow, and they're right in their own way. But then this happened again and again and again for days, for weeks, for months. And this experience then kind of made me realize how climate change is actually affecting small parts of our daily lives. And then that made me thought that yes, this is something no more a mythological concept as such, and yes, it's no more something which I'm just reading in an environmental science book, and this is how what I'm experiencing, to the point that some days of the summer of 22 became just unbearable. So then how are we tackling it, or how are we going about it? If I speak from a scientific community point of view, there are mainly 2 broad ways in which we are kind of tackling climate change. The first one would be we kind of shift completely from fossil fuels to renewable energies. That's a very active field of research. But then till the time renewable energies reach the level of providing the rising energy demands of our population, it will still take many decades, and for all those decades there still will be emissions of CO2, which is a primary contributor to climate change. And that is where comes the second round of, or the second route of tackling climate change, which is carbon capture, and which itself has its different ways of capturing CO2 from industries, directly from air, and many other areas. Now, whichever route we go in, one very central part which is an important key in both the routes are materials. People have been researching in different classes of materials, be it 2D materials, 3D materials. People have been working on graphene, activated carbon, zeolites, different framework materials, a huge range of materials. But the specific class of nanomaterials which I am going to focus in today's talk are metal organic frameworks, or MOFs in short. And before I proceed further to discuss about MOFs, just a quick question to have a rough gauge. How many of you have probably come across the term MOFs, or metal organic frameworks? Okay, not bad, some of you. Okay, so for those of you not aware, a MOF, or a metal organic framework, consists of a metal part, a metal node, an organic linker which is connected together in form of a template or a network which we call topology, which then gives rise to these beautiful nanocrystals. Over the last 2 decades, MOFs have found themselves to be very useful in a wide range of applications like gas storage, catalysis, drug delivery, sensing, and many more. And a big reason behind it is that it consists of a very interesting combination of a wide range of properties. For example, it has really high surface areas, and to give a context of this, just 1 gram of a MOF can cover an entire football field. So it has surface areas ranging up to 5,000 meters square per gram. It has pore sizes which enables to trap different gases like say CO2, N2, and therefore put them inside their cages. It also has tunable chemistries. For example, if you by combining these metal nodes and organic linkers and in different topologies, really infinite amount of MOFs can theoretically be made. And all of this has led to over 100,000 experimental MOF structures being synthesized over the last 2 decades. And with the advancement of computational techniques and simulations, we also have millions of hypothetical structures which have been generated in silico. Okay, so far so good. We have millions of materials, we have computational power, we screen them, we try to find the best materials from the screening. But this also creates a dilemma for this entire MOF community. The dilemma being that on one hand, at present, we have so many materials that even if we have the fastest computer on the planet, we cannot end up screening millions of these materials. But on the other hand, as I mentioned, theoretically an infinite amount of MOFs can be actually be possibly made. So that is what is represented in this graph. I will come to the technical details of this graph in the subsequent slides, but for now what I want you to take from this slide is that this kind of represents these gray dots which you see, kind of represents unexplored areas of the material space, the material MOF chemical space, which we haven't explored that much, but a MOF can possibly exist over there. So then how do we create a balance between these two contrary search? Because a holy grail of material science has been to find the most optimal material for a given application. And therefore we decided that we need to take a different approach from the brute force screening, and we decided that we're going to take a diverse sampling of this MOF chemical space and create a diverse database of materials, screen them, and then find, instead of finding the best one material for a particular application, we want to locate what is the most promising regions or the best regions of the space which contributes to a material being good for that application. And once we find that interesting region of the MOF space, we can then populate that area with different techniques. And for that, we developed a computational workflow comprising of different techniques using molecular simulation, machine learning, and material design. I'll just briefly, I don't want to go into too many technical details, but just briefly give you a glimpse of what this MOF world might look like. So for example, here you are seeing the building blocks of these materials, or what we also refer to as the genes of these materials. So on the left hand side, you see some of the metal nodes. On the right hand side, you find some of these organic linkers. Then you can functionalize the organic linkers with different functional groups. And this whole range along with networks and topologies create a very high dimensional space. And with this, we created an in silico database of around 20,000 materials. And once we created this database, we wanted to visualize that if at all there was a material space or a universe of materials, how would it look like? But then it's a 200 or a very high dimensional space. So how do we visualize the 200 dimensional space? We can't make sense out of it, right? So what we did is that we used machine learning techniques to reduce this high dimensional 200 space to a 2-dimensional space to get a rough idea of if this universe of materials did exist, how would it look like? And what you can see as a glimpse over here is that each point in these graphs represents a material. Wherever you see a colored dot in terms of the MOF components, so for example, this represents the metal part of the MOF, this represents the organic linker chemistry of the MOFs. So the colored dot represents those areas which we have sampled from the known chemical space of MOFs, and in each plot you will find there are some gray areas that represents the unexplored or the relatively less explored areas of the morph material space. It's like, imagine you have this galaxy and this universe of different materials where certain materials exist, but we have still not set our foot onto it. So to me, the way I see it is that, hey, this is one aspect of morphs, like say for example the geometrical properties of materials which we have explored quite well, but the other areas we need to work harder. For example, metal chemistries we can do much better. And in this way we iterated, we made many iterations, we tried to see what is it that we can improve the diversity, and then we developed different quantifying techniques to show how adding our materials improved overall diversity of this morph community. And I'm not going into the maths of all these things, but this is just to see that how diversity was a tool in this particular aspect. Okay, so far so good. We are visualizing, we are having this database of materials, but then we thought, now that we have these materials, can we use it for some practical applications, or does it just get limited to a theoretical concept? So one of the applications which we are really interested in was carbon capture, and what you see again is a small example of 4 of the metal clusters which we used, and you will find the histograms of different materials, and in each plot you will find a blue line. The blue line basically represents the reference line for zeolite 13X, which is a commercially available carbon capture adsorbent which is used in many industries, the oil and gas industries, the coal power plants industries, and we wanted to see that if any of our material is better than the current existing material. So you can see, for example, morphs made of these kind of metal nodes are practically useless for our applications, because hardly any material or a very few percentage of materials are better than the benchmark. But then morphs from these materials, we have quite a few interesting candidates. And that is how we came to also find another similar candidate which was not in our initial list, but we went back to this plot and we tried to say that, okay, if this is an interesting region of the space and if we find a dot over here, then there should be some materials around here which will also be interesting. And that is how we also came to find this particular metal node. And we extended this to even other applications. We extended it to hydrogen storage, we extended it to methane separation, and like the thing which I liked most about this thing was like my initial aim was not to find best materials for hydrogen storage. I was only focusing on carbon capture. But since we did a diverse sampling of this entire space, some parts of the space contributed to materials being good for carbon capture, but some parts of the space contributed for a material to be good in hydrogen storage. And those two material lists may not necessarily overlap, like the materials good for this application may be absolutely useless for this application, but the diverse sampling helped us locate that region. So if I just summarize the overall computational workflow which we have used over here, we start with the genes of a material, which is the building blocks. We build a material from there. We then create databases of these materials, experimental, computational. We screen them using different computational techniques and try to find out which are the promising materials for, say, an application like carbon capture. And then we also now going one step ahead and testing these materials in real industrial conditions and trying to see that which of the materials actually last in the final industrial cycles. And in order to help us in our aid to search this huge infinite dimensional space more effectively, we are taking the help of machine learning techniques where we're trying to map directly from this genes of a material to the final application and the property, so that we can find out and say that what is it in a material. Is it the metal chemistry? Is it the linker? Is it the particular bond between the metal and organic linker? Or is it the orientation between the 3D structure of a morph which makes it good for a material? And that helps us to enable to design even better materials for next generations. Thus, in this study, what we show that is even if we start with a relatively small but a diverse set of materials, we can still obtain promising material for different applications. And once we locate that interesting region of that material space, and I keep repeating this word material space so that we have that visual thing in our mind that we are setting our foot into unexplored regions, we can then explore around those interesting regions using different techniques, mathematical and physical techniques. And this thus also helps in a broader goal of materials discovery, thus extending it to many other classes of materials also. And with this, I'd like to thank my supervisor, Professor Baron Smith at EPFL, and my lab members, and all my funding agencies, and last but not the least, all of you for your patient hearing. Thank you.
Thank you. Thank you, Saradip. Now we
you can just move there and ask. Yes.
So I also I was something about machine learning and the morphs for the application. My question is, so the machine learning exists for the morphs. So for the simulation of the morphs has existed in the database, or so can we got a principle for the application? For example, for water stable. For the CO2 absorption with the water stable morphs. So my question is that the machine learning is from the existing morph or the new, or can predict new morphs?
It's both ways, I would say. So one thing is that for existing morphs, for like experimental structures, we try to predict, like for example, in the example you say, like say for example, you have a water stable, like whether a morph is stable in water or not, that can be predicted for experimental structures, and I believe that is a thing which has, I think, is being done now at quite a good level. And the next level which we are trying to solve is, can we predict structures? Like for example, using concepts like inverse design. Like for example, can we design structures and predict a structure which we know a priori will be good in water or stable in water or in a realistic flue gas condition? Okay. Was that your question? Sorry.
Okay. And then we can synthesize the predicted morphs for the experimental to verify the properties.
Synthesis is a thing which goes parallelly, yes. I mean, of course, I wouldn't say that all the morphs which we predict theoretically get synthesized. Of course, I wouldn't say that. But yes, there have been many examples also in recent years where many of the morphs which have been theoretical. So typically what we do is that maybe from millions of structures, we would probably give my experimental colleague 50 structures, that these 50 are probably the best out of 5 million structures. And then the experimentalist, maybe out of those 50, maybe 30 of them will be discarded because, I don't know, expensive linkers or some other issues and all, maybe only a few of them will be made. So if you are speaking of the percentage, yes, the percentage is few, but then it helps us just navigate the space better, I would say.
Oh yeah, thank you very much.
Thank you, Vagresh.
Hi, also thank you for the presentation, and what I particularly enjoyed is your use of visualizations. In particular, you were using TSNE, dimensionality reduction method, to visualize basically the, let's call it the material space, right, to visualizing. Did I understand correctly that you were also using this visualization to identify new areas where you want to search for new materials that have not been discovered before?
Yeah, so for example, I showed about, so there are different dimensionality reduction techniques. So we have principal components, we have TSNE and all. So since I mentioned that we have this, say, a 200 dimensional space, so especially in the case of PCAs, once we get a coordinate of a particular space with respect to a particular feature of a morph, we can locate what is the nearest other morphs which are near to that point, because similar materials will be located similar in the material space. So that was the philosophy of finding new materials. So probably something lying out there which we didn't sample first, and then we take out from there and make a material out of it.
I find this very fascinating. I've also used UMAP before for similar
purposes. UMAP is one more technique. I haven't used it, but yes, that's also one more good technique.
A question that I have, which is like a common limitation of TSNE or UMAP in that sense, is that they are not necessarily distance preserving in the original space. So
you are correct.
I'm also asking myself if one can today rigorously argue about the search method when we have no guarantees that we're actually preserving distances in the original space.
Yeah, that's a really interesting point. I mean, yes, of course there is no guarantees, because the TSNE and UMAPs are also based on statistics. whereas principal components on that aspect preserves the global variances much more. So from a principal component point of view, I've shown only the Tesla maps over here, but we also had a principal component analysis, and along with that we also what we call, or I would term it as chemical intuition, which like you combine your chemical intuition along with the machine learning data to come up with that morph. Like, okay, so what kind of metal atom, what is the oxidation state, what are the type of connection points, what kind of linkers, what are the other properties, and to have a similarity index. So I would say it's a combination of the machine learning techniques and the scientist's own intuition, which would probably go forward to select a material.
Okay, very cool. Thank you so much.
Next, please.
Thank you for the talk. My question is, does the machine learning able to predict some of the limitation on like the synthesis? Like for example, like, you know, like some of the structure maybe is thermodynamically unstable to be synthesized, that like the cavity is just too large. And if it doesn't, is there anything that you can do in the future so that the machine learning is able to predict like those limitations, so that, you know, whatever is being predicted on the material, it is actually possible to synthesize it experimentally? Thank you.
Yeah, one work which was very recently published was actually in this area where natural language processing tools, like for example, something which Google and Microsoft uses, in those lines, all the experimental data from the last 2 decades were actually used as training in a group, and they tried to predict what is the thermal decomposition temperature for the morphs. So for example, till what temperature are the morphs stable? So this was, I think, in this field quite a good leap. So it, of course, it won't give you the guarantee that, of course, if a morph is stable, it will be made for sure. That will still take some time, but at least there are efforts being made, and I'm hopeful that it will be going in that direction. At least till now, we can give a rough indication that, okay, till what range of temperature is this morph stable, and when does it start to degrade? And then maybe study in more detail the few character structures which we are not very sure of.
Thank you.
Any further questions from the audience? Perhaps I can ask your question while the audience consider more. My first question is general. You are a PhD student.
Yeah.
So far, what do you enjoy the most about your PhD, and what do you dislike the most?
Okay. I think it changes with the year of my PhD. Every year the likes and dislikes changes, but yeah, if I kind of sum up, I mean, I think PhD in a way helps. So I'm coming from a chemical engineering background. I did my bachelor's and master's in chemical engineering. I worked in industry. So I've always been in a more application oriented field, like always in a thing. And I wanted to experience this other side where you can actually use creativity and combine it with applications to have a greater degree of freedom. So this is one thing I really enjoyed, that, for example, the example with visualization. So for example, finding something through a mixture of creativity and sense of application. I think this is a freedom which academia to some extent gives us, and I, at least in my PhD, I really liked it. Dislikes, yes, of course there are many. Of course there is probably, I'm working with some statistical thermodynamics, so we often use these error bars, right, in many of our measurements. And I find that, yes, the uncertainties are so huge sometimes in whatever things we do, that maybe we work probably many years, maybe something comes out, maybe something not comes out, especially in my first or second year of PhD, that sometimes got me derailed or demotivated. So that was the hard part, I would say. And yeah, so I'm lucky that somehow I managed it.
Okay. Okay, good. So the thing you enjoy the most of your PhD is you are able to really work on projects with application perspective that use fundamental knowledge, fundamental creation. Yes. You can solve really grand practical challenges.
Yes.
And the thing you dislike the most are some actually technical limitations, some frustrations in the more detailed process. Okay. I have one more question for you. You are doing simulations and machine learning. Do you fear your work has to be sooner or later combined with experimentalist work? And how are you going to achieve that?
Yeah. So yeah, this is something which definitely goes on in my mind. And for example, in one of the current studies I'm doing, actually, one of my experimental collaborators actually was able to synthesize one of the materials which we made. And of course, it was a difficult task in general, like combining simulations and experimentations, because you need a lot of trials and stuffs, and things may not always go well. It is something I would say, I won't say it's a fear, but yeah, it is definitely something which I keep in my mind when I'm working on my future projects, or if I'm thinking of starting a project or working with project, that how to kind of link it to an experimentation. Sometimes it doesn't work, like probably because of different factors. But yeah, there was like few examples where it did work, and yeah, that was good.
Okay, thank you very much. Is there any final question from the audience? Okay. If not, please join me to thank Saradip one more time for his presentation.
Thank you.
And now it also gives me the pleasure to introduce the next speaker and also the final speaker of the session, Una Trivanovic. She is also a PhD student from Switzerland, from ETH Zurich. She's from mechanical engineering and will be talking on some science on combustion. So Una, the floor is yours.
Great, thank you very much for the kind introduction. So as you just said, I'll be talking about my work using enclosed spray combustion of jet fuel as a surrogate for aircraft emissions. So we'll be staying on the topic of climate change and nanoparticles. So when you think about aircraft emissions, chances are what you think about is carbon dioxide. And of course, this is an important component of the emissions. However, it's not the only thing that comes out of an aircraft exhaust. Another thing that's very abundant is water, actually. And the focus of my research, these soot nanoparticles. And a few things happen in this case. For starters, the soot can actually act as a nucleus for the water to condense, and this is what forms contrails. In addition, the soot can act as a direct absorber of sunlight and thus warm the atmosphere in this way. So it's important that we understand what these particles are and how they interact. This is so important, in fact, that these non-CO2 radiative forcing effects account for nearly 2/3 of the net radiative forcing for aircrafts, highlighting the importance of understanding these effects, not just measuring the CO2. On the other hand, these particles can also have a negative effect on our health. So when they're emitted around airports, in particular, aircraft emissions tend to be very small compared to, for example, the particles from a diesel engine, and these have an outsized negative effect on human health. So it's important that we understand what we're emitting. So to understand a little bit more about what these particles are, here I have a small cartoon of what this looks like. It's this irregular particle. They're based on carbon. And we have these primary particles that make up the larger aggregate. Within that, there's also a nanostructure to these primary particles. And we learned about graphene earlier in the day. You can think about this as layers of graphene surrounding a more disordered core in the middle. And what this looks like will really influence how it is able to absorb light and what its direct radiative forcing will be. In addition, we need some way to measure the size of the whole particle. Because it's irregular, we can't attribute a direct measure of the diameter to it. So what we do is we use equivalent diameters, and what's often used in the field is the mobility diameter, so the size of a sphere with an equivalent electrical mobility. Next, we want to quantify in some way the chemistry of the particles, and for this we often use the organic carbon to total carbon ratio, and you can think about this as the organic carbon are the volatile or semi-volatile compounds that are condensed on the surface, and the total carbon is everything, including the solid parts that we have. Finally, one thing that's very important, but actually very rarely measured in the field of aerosol science, is the specific surface area. This often isn't measured because it's very difficult to get a large enough quantity of particles to do what we call nitrogen absorption, but it's very important because it's one of the most important metrics for quantifying the toxicity of any kind of nanoparticle, including soot. And so in my work, what we've been doing is we've taken real jet fuel that you would use in an airplane, we've turned it into a spray, and we combust it in a quartz glass tube, which allows us to carefully control the conditions within the tube. And we can also change the conditions through what we call the effective equivalence ratio, which is just a measure of how much fuel and oxygen there is in the mixture, with 1 being stoichiometric conditions and greater than 1 meaning you have excess fuel. And so by doing this, we're able to produce particles which have the same parameters, which I've just pointed out, the primary particle diameter, the OCTC ratio, in the laboratory as we do in real aircrafts. So now to understand how these particles grow in a very simple flame, here we have a premixed flame. So first we start off with what we call inception. This is where the particles go from the gas phase into the first solid components, and these are just a few nanometers in diameter. As they continue to travel through the flame, they go from a few nanometers to a few tens of nanometers through surface growth, where chemical reactions with the surface grow the particles. We can also have aggregation, where if two come together, they chemically bond together to form these necked dimers. Then, as we continue farther, we get agglomeration. During agglomeration, the particles simply by chance collide into each other and they form a weak point contact bond, and at this point they can grow considerably to as much as hundreds of nanometers in diameter. And in the final phase, we might have oxidation. This is the only phase where the particles actually decrease in size rather than continuing to increase in size. And so this is because of excess oxygen surrounding the flame becoming entrained, and then it oxidizes away to either a smaller size or completely into carbon dioxide. So this is a very simplified model, but in reality, in aircrafts, in engines, we have a much more complex turbulent flame, and so this is what we researched. So in our complex flame, we took a sampling probe and inserted it at different heights above the burner, which is analogous to the residence time that it's experienced in the flame. And so this let us measure how it's evolving. So here we see that as we increase the height above the burner, the mobility diameter increases steadily throughout the flame. And because it's never decreasing, this suggests that we have minimal or no oxidation. Then we looked at the primary particle diameter, and we saw that this actually did not change from the smallest height above the burner that we investigated, 5 centimeters. It stayed about constant throughout the flame. So to understand why this happened, we also did some computer simulations. And first, we see in the blue line here that our simulations matched excellently with the data. And similarly, with the primary particle diameter, it was in good agreement with our experimental data. So what do these experiments show us? The simulations say that in this range where we were not able to measure experimentally, we actually had single primary particles that are significantly growing through surface growth. And so as they grow in size, they reach a point at about 5 centimeters where we have these aggregates of just a few primary particles. From here, agglomeration takes over as the primary mechanism, and they continue to grow into these large mobility diameters, but the primary particle size stayed roughly the same. Next, we wanted to understand something about the nanostructure within those primary particles, and we did this using Raman spectroscopy. So first, to put this into context, we have Raman spectroscopy from graphene, which again we talked about today. So graphene is a very ordered crystalline structure compared to the soot. And here we have the D band, which you can think about as the amount of disorder in the system. The G band, which you can think about as quantifying how graphitic or ordered the structure is. And also the 2D band, which we won't focus on so much. It's more a function of how many layers of graphene there are. If we now go to the few layer graphene, we see a very small D band and a large G band. So this suggests that we have a very ordered structure. When we go already just to graphene oxide, where we have more defects in the system, this D band significantly increases, and we can use this D to G ratio to quantify approximately how disordered it is, and we go from 0.06 up to 0.9. And also in the literature, we have these carbon black nanoparticles, which are quite similar to soot, and they're actually a commercial nanoparticle, which is in car tires. In our work, we saw this characteristic D to G peak, and at the lowest height above the burner that we investigated, we had a D to G ratio of about 0.8. And as we go through the flame to higher equivalence ratios at 10 and 25 centimeters, we see that this is increased a little bit. And at the exit, when it exits the reactor, it goes up to 0.9. So this suggests that the particles are actually becoming more disordered as they travel through the flame. And so this could be because of reactions on the surface. And importantly, when we compare this now to real aircraft soot from the literature with this dotted line and the shaded area representing the different conditions that an aircraft can operate at, we see that we're able to reproduce quite well the nanostructure that we see in the Raman spectroscopy between real aircraft soot and our own laboratory soot. So up to this point, I've just shown you for a single equivalence ratio or a single combustion condition what the results look like. So here is that same one that we've seen, the same spectra you saw on the last slide, and the distribution of primary particle sizes, because it's not just one. But we can also change the combustion conditions. So if we increase slightly the equivalence ratio, we see virtually no change in the Raman and the primary particle size distributions. If we continue, however, to equivalence ratios of 1.73 or even 1.88, now we see a significant shift in the primary particle sizes. So these leanest conditions here are similar to aircrafts, while something like the 1.88 with a 23 nanometer primary particle size compared to 13.6 nanometers, this is more like what you might find in a diesel engine. Now, interestingly, across this same range, we see that the D to G ratio, or the amount of disorder in the particles, has decreased significantly. This means that the larger primary particles tend to have a more ordered nanostructure. And if we compare this directly with the median primary particle diameter, we see that we have an excellent correlation between these two. And this is important because this determines how it interacts with sunlight, and it could be that these more aircraft-like particles do not absorb as much sunlight as the more diesel-like particles, for example. And other work has actually shown in a completely different flame, they disaggregated the particles based on their size, and they saw the same trend. So this suggests that this really is a function of the size of the particles and not just the different conditions that we used in the flame. So now we want to understand also how the changing conditions affect the mobility diameter. So I'll point out that these 2 points are the lowest points we saw on the last slide, and we actually looked at some even smaller equivalence ratios here. So by increasing the equivalence ratio, we were able to dramatically increase the mobility diameter from this extreme case of an equivalence ratio of 1.29. We had an mobility diameter of about 15 nanometers, and we increased it by about an order of magnitude to 160 nanometers at this highest equivalence ratio. Interestingly, in this range, we saw a very constant primary particle diameter. And so this is interesting because we've grown the mobility diameter, but not the primary particle diameter, which only started to grow off the end of this graph when we increased the equivalence ratio to 1.7 or higher. And so this is important because what it tells us is that the mobility diameter increased because we had a higher rate of nucleation, so more particles with the opportunity to run into each other through agglomeration, but surface growth was not acting importantly in this range. And so here we have some images of the particles, and you can see that they're quite similar. And importantly, the range that we're in here of about 14 nanometers falls squarely in the range of aircraft soot. So now the last thing, and the most important thing that we wanted to look at, was this specific surface area. So we used nitrogen absorption to measure this, which also allowed us to look at the pore size distributions. So at the lowest equivalence ratio we showed here, we had a specific surface area of about 160, and looking at the pore size distributions, we see a relatively small number of pores, which at the small pore widths begins to increase. as we increase the equivalence ratio a little bit, we see basically the same trend in the pore size distribution, but we have a slightly higher number of pores, which of course results in a higher specific surface area. If we go to even higher equivalence ratios of 1.46 or 1.59, now we do not see the same trend anymore. We have an increase in particles in the range of 2 to 4 nanometers. And this is important because now we have a significantly higher specific surface area, and this higher specific surface area can in turn lead to higher toxicity of the particles. And we want to understand what caused these pores. So one thing that could explain this is the difference in temperature. The closer we get to stoichiometric conditions, the higher the temperature will be in the flame. And what happens at high temperatures is that oxidation, if any is occurring, occurs quite fast, and carbon is removed quickly from the surface of the particles. At slightly lower temperatures, oxygen is able to diffuse farther into the particle and remove carbon from the inside. And this could explain why we have this porosity forming at the higher equivalence ratios compared to the lower equivalence ratios. And I'll point out that in our work, this higher equivalence ratio of about 1.59 matched very well with aircrafts at takeoff, or what we call 100% thrust. So this could indicate that we would see the same phenomenon in aircrafts at these conditions. So now to summarize what I've told you, what we did was we took a spray combustion of real jet fuel, and we used this to recreate aircraft-like soot in the laboratory, allowing us to get a better understanding of this in a laboratory setting. Thanks to this device, we were able to measure the specific surface area of soot and also recognize that there are these internal pores that are contributing to the surface area. And finally, we also observed that as the primary particle increases, we also had an increase in the order in the nanostructure, which will affect, in the end, the interaction of light of these particles. So with that, I want to thank you for listening and welcome any questions.
Thank you, Una. Now we have a short Q&A session moderated by Dr. Yen.
All right, now it's time for Q&A again. Yes, please.
Hello, good afternoon. Thank you for your interesting talk. I'm just interested when you mentioned about the flame turbulence. Can you expound on that? Did you consider the air turbulence around the flame? Because let's say during the takeoff, there should be an air turbulence around the combustion region, which should affect the formation of the soot, or let's say the air turbulence could affect could result to disintegration of bigger particles. Yeah, if you just consider that air turbulence, if you already mentioned about the flame turbulence, and if you compare, let's say, the standard Reynolds number of the flow regime around the furnace, which you recreate in the laboratory setting. Thank you.
Yes. Can you hear me? Yes. So, of course, the turbulence is very important, and the reason I brought this up is because a lot of the fundamental research on how these grow is done in these laminar premixed flames, which are in a lot of ways an oversimplification of the real system, as you said. Our system is also very turbulent. We haven't measured exactly the Reynolds number, and there are quite a few other differences compared to a real aircraft, of course, so it won't be one-to-one, but both systems will have turbulence, and I think the important thing about the turbulence is that it will give you heterogeneous nanoparticles, so you don't have just one. This is why we also had the distribution of primary particle sizes, for example, rather than one single primary particle size, because they're forming in different regions of the flame, so we really have to talk about the distribution of particles as opposed to just one. And so when we're comparing to the literature on aircrafts, we're comparing to the whole distribution of particles, not just one single particle.
Thank you.
All right. Any other question? Actually, there are questions. There's one question from the online audience. Let me read it out. What numerical model equations was used for considering the chemical reaction, fluid flow, and also particle tracking?
Yes, so we used a model called discrete element modeling. And so what this does is basically we have the particles from after they've become those solid particles in the inception phase, and we track how they undergo surface growth and agglomeration through the flame. In this model, we do not account for the fluid flow. We just look at how the particles are moving through random chance through a small space, and then extrapolate this through to get average quantities for the particles. And when we run this several times, we can see how the distribution would occur, because each time it's a stochastic model, and so it's not exactly the same each time.
All right. Any further question? Perhaps, oh, yes, good.
So also, thank you for your presentation. I also understand this is probably then continued research, but you were also talking about, for example, toxicity, and I think your results, I guess, give way to certain actionable measures that we could implement, for example, at takeoff and so on. So do you think that from these results you could give some actionable advice to how, for example, flight traffic is executed?
Yeah, of course, it's a very complicated system. I guess there are a few easy things that you can do. One is avoiding idle in aircrafts, in airports, which would improve the number of particles that are accumulating just unnecessarily. There are some things that you can do using alternative fuels. Some fuels tend to produce fewer particles than others, but it's important in this case to look at the trade-off, because of course particles are not the only pollutant, it's just one of them. But some newer fuels do tend to produce fewer particles, so you can remove it this way. I've also seen some ideas around spraying water vapor essentially at the exhaust to try to remove the particles from the local air. And another very important thing that I hope can come out of this research is we still need to understand more about what causes contrails to form in some areas and not others. And so there is work being done out there using the characteristics that we're able to gather about what the particles are to put it into models, which will then help us to understand why in some cases we have contrails and some we don't, and maybe we could modify flight paths to avoid contrail heavy regions.
Very interesting, thank you.
Okay.
So
yes.
I wanted to ask, in your Raman spectra, when you have the graphene versus the graphene oxide and then you again heat up even more, in that case, there is two things happening. One is the structure is changing, as from graphene you go to graphene oxide. The other thing is it is also getting aggregated, right? So you have a heterogeneity in your system. So then how do you structurally assign the Raman spectra? Is it because of the basic structural change, or is it coming from the heterogeneity or aggregation?
So in the example that I gave, it was not pure graphene. It would have been few layer graphene and an aggregate of it. So there was still that small D peak. It wasn't a perfectly crystalline structure, because I guess it was a graphene that was made with methods that were not super precise. And so in all the cases, yes, it's a heterogeneous measure of an average of what you're seeing. And typically also when we do these measurements, we'll measure at several different points in the powder so that we understand how it's varying from position to position. So it's not as precise as it would be in a purely crystal, like if you're looking at a single crystal, but it still gives us some idea when we are comparing spectra within similar materials how it varies.
All right. If there's no more question, please allow me to ask a few. Number one, actually, it follows what the last audience has asked. You're following the carbon formation, and you mentioned a number of parameters that affects carbon formation. I wonder, what is the relationship between the chemical structure of the fuel? We can have normal jet fuel, but now people are also talking about the biojet fuel and other types of jet fuel. When the chemical structure of these fuel change, how does that affect carbon formation?
Yes, this is very important, and this is something that's an active area of research in the field right now. So one important thing is traditional jet fuels tend to have a high aromatic content in the fuel compared to some of the biojet fuels which have less of it. So these aromatic compounds in the hydrocarbons that are being combusted, those tend to be precursors for soot. And so fuels that eliminate that tend to have fewer soot emissions, but not all biofuels that are being looked at have fewer soot emissions. So it really is important to look at them individually to make sure that you're really reducing that with the biojet fuel, because sometimes they're not optimizing for that.
Okay, thank you very much. My final question is general. I asked the first speaker about present in PhD. I want to ask you into the future. After PhD graduation, what are you expecting the most, and what worries you the most?
I guess there are a lot of unknowns, so it's easy to answer that what worries me the most are simply the unknowns. But I'm excited about the opportunity to have some more freedom to decide in my own directions. And one thing that I've really enjoyed in my PhD at ETH, where I have the opportunity to supervise bachelor's and master's theses. And this is something I really, really enjoyed. And I think that in the future, I really look forward to having the opportunity to continue working with other scientists and coming up with projects and working on things, with people above and below me, I guess.
Okay, thank you very much. Now there's no more further question. Please join me to thank Ona one more time. Now let me hand over to MC.
Thank you, Dr. Yen. Now we would like Dr. Yen to present certificates of appreciation to our presenters. So first up, we have Una Trivanovic. You can open it up. Thank you. All right, and next up we have Saradeep Majunda. So we have now come to the end of today's Young Scientist presentations. So thank you to our Young Scientist and Chair of the session, Dr. Yen. So this evening there will be a short talk on how science can support the Sustainable Development Goals by Mr. Caleb Hanford from the United Nations Development Programme, UNDP Singapore. This will commence at 6:15 p.m. at this theatre. After which dinner will be served at the campus center at 6:45 p.m. Where you guys can continue to network and interact. For those who have signed up for the SUTD campus and lab tours, appreciate if you can please quickly make your way to the campus center. For now, we will adjourn for a short break, and I'll see you back here for the UNDP talk at 6:15 p.m. Thank you. Will commence at 6 p.m. instead. So if you would like to join a UNDP talk, it will start in about 5 minutes' time. Thank you. All right, good evening, everyone. We would like to invite Mr. Caleb Hanford, advisor from the United Nations Development Programme, UNDP Singapore Centre for Technology, Innovation and Sustainable Development, to talk about how science can support the Sustainable Development Goals. Mr. Hanford has led innovation and digitalization efforts globally and has also advised governments around the world. He is the co-chair of the Digital Infrastructure Task Force that is part of the World Economic Forum's G20 Working Group on Smart Cities. There will also be some time for a Q&A session, which will be moderated by Prof. Benjamin Keshaw from the Lee Kuan Yew School of Public Policy at the National University of Singapore. Mr. Hanford, please. Thank you.
Thank you, everyone. It's a real privilege to be here, and I know you've all had a very long day, so I'll try and keep this quite short as well. Let's see if this works. Perfect. So a little bit of context about us. So the UNDP, the UN Development Programme, is one of the largest delivery organizations of the United Nations. We work with countries to expand people's choices for a fair, sustainable future, to build the world envisaged by the 2030 Agenda, and obviously keep people and planet in balance. We have about 170 countries around the world, country offices around the world, about 20,000 staff. And as I just mentioned, I work at the UNDP Global Centre for Technology, Innovation and Sustainable Development, which is a collaboration between UNDP and the Government of Singapore. Perfect. So what are the SDGs? So some of you may already be familiar with this, but the SDGs are 17 goals adopted by the United Nations in 2015. They are a universal call to action to end poverty, to protect the planet, and to ensure that by 2030, all people everywhere enjoy peace and enjoy prosperity. The 17 SDGs are all integrated and interlinked. They recognize that action in one area will affect outcomes in others, and that development must balance social, economic, and environmental sustainability. The goals are also unique in calling on all countries, whether rich, poor, big, or small, to work towards achieving them by 2030. And they also recognize the role of all industries, sectors, and actors in their success as well. So the SDGs were a very ambitious target ahead of the pandemic, but the challenge now is even greater. We saw that global poverty increased in 2020 for the first time in 2 decades, and growing global turmoil has led to unprecedented reversals in human development progress in about 90% of countries around the world. We're seeing human development reversing for a second consecutive year, which actually constitutes a backside in human development that we've never ever seen before. And this has led to what we at UNDP are calling a new uncertainty complex. So humanity's biggest challenges, the climate and biodiversity crises, inequalities, political polarization, as well as technological upheavals, are all piling up and feeding off each other. And this is all happening at a speed and a pace that we've never ever seen before. But we've also seen opportunity happening in this kind of uncertainty. And science has been a really great demonstrator of this. Over the past few years, during the course of the pandemic, we've seen science evolving in real time and global collaboration happening when the stakes couldn't be higher. And the unprecedented success of the vaccine program and development has been a really kind of key demonstrator of this as well. But these unprecedented challenges also need a truly global and truly comprehensive response. And you can see here just a snapshot of the range of different scientific and broader interventions needed just for tackling one area, to reduce agricultural emissions, from better diets to reforestation. Incidentally, actually, this graphic is taken from the UN Global Sustainable Development Report, which is prepared by an independent group of scientists. It presents an objective assessment of where we are falling short and what needs to be done. And actually, the next iteration of this report will be published later this year. And for those of you who are working or living in the Gulf region, there's actually a consultation happening on this next week. What we know, though, is that science is a really important resource, asset, and partner for development and for the SDGs at a local, national, and global level. And you're all incredibly important partners on this journey. And I'd like to explore that in the course of my presentation, focus on 3 big points. The first is the importance of looking for synergies. It's often quite easy to end up with our head down, focused on a particular solution or a particular problem, and to get stuck working in silos. So each node that you can see on this chart identifies one SDG, and the size of the node is proportional to the number of scientific publications relating to that SDG. And each color also indicates a particular cluster. You can see on the top left, natural environment, infrastructure, and people and society. The lines that connect SDGs that are studied by a number of research areas. So the thicker the line, the more research areas are related to both connected SDGs. For example, SDGs 13 and 15 on the left-hand side share a number of research areas and publications, whilst SDG number 4 on the very far right shares only a small number of research areas and publications with SDG number 10. Incidentally, SDG number 11 in the middle is actually the only one that's connected to all 3 of these research clusters. What we can see with a lot of science in relation to the SDGs is a focus on technological responses to particular challenges. For example, access to energy, or SDG number 7. But often there's a lot less research focusing on addressing the complex and underlying social issues, challenges such as deprivation, inequality, and conflict. And also less research looking at how technologies link into these complex issues as well. I'll provide a quick example. So UNDP, we've been collaborating with a number of global research actors on the STRINGS initiative. So STRINGS stands for Steering Research and Innovation for the Global Goals. And one of the findings was that research related to building science, tech, and innovation capabilities, so SDG number 9 about industry, innovation, and infrastructure, is often carried out most frequently in connection to research on technology solutions related to a small number of SDGs. But it doesn't engage with the complex underlying issues of quality education, reducing inequalities, tackling poverty, or building strong institutions. And then this creates issues in turn with investigating the synergies and tensions between science and the SDGs. And this is a really systemic and a really structural challenge. We need to focus on research areas that connect to several SDGs by funding more research explicitly investigating these tensions and synergies. And we also need to be connecting and funding research on the fundamental issues of deprivation, inequalities, and conflict with research on these more technological and technical solutions. There's actually a good example here from Singapore. So Singapore is actually one of the first countries during the very outset of the pandemic to invest in funding a digital contact tracing solution. And they also open source this, so the code is available for other governments to borrow and to take forward. And there's a risk, obviously, when they launch this, that there would be a risk of a reliance on a single technological solution. But when they launched a solution called Trace Together, they actually included this quote in the press release, highlighting that Trace Together was not a single solution for the pandemic or a single solution to tackling COVID-19. It was part of the broader public health toolbox and a complement, an augmenter, and an amplifier. And this also parallels the kind of broader research thinking that we need to be doing in SDGs, looking at the kind of broader picture and not focusing on single solutions. The next point I want to make is the importance of thinking at a more global level as well, wherever possible. And there's actually a question of alignment, with research often more broadly not aligned with the SDGs. The majority of research in the countries that dominate the global research agenda, so higher income countries, is often not related to the Sustainable Development Goals. And when it is, it's often quite skewed. So 60% of the research in higher income countries in SDG context is actually related to just one goal, SDG number 3, around good health and well-being. And often this is even focused on diseases that are most prevalent in these higher income countries. If you compare this to low income countries, like you can see on the graph on the left-hand side, those who are facing the most SDG challenges, and the vast majority of research in these countries is actually relating to the SDGs. But their influence on the global research agenda is drastically asymmetrical, as they produce 0.2% of all world of science research that's recorded. So we need to rebalance this asymmetry and look at providing platforms, support, and true collaboration. And this is not an isolated challenge either. We see that the risks and challenges of technology and innovation, so things like automation of jobs, inequality, discrimination within data, and fair intellectual property rights, often disproportionately affect developing countries. And also things like venture capital and other funding penetration is also highly variable across regions, whilst the intensity of research and development funding, including patenting rates, is often strongest in many higher income countries. You can even see this in publication rates. There's often a handful of scientific and technical journal articles per 1 million people in least developed countries, compared to more than 1,000 journal articles per 1 million people in higher income countries. And often we also see that most innovation literature focuses also on just a handful of low income countries as well. So this kind of disparity is from top to bottom. But we also need to be building data capacity to understand progress against the SDGs. When we work in many countries, often one of our big partners will be statistical authorities and national data departments, and many country statistical systems often struggle to provide data on the SDG indicators. There are serious gaps in assessing priorities, in assessing funding, and assessing country-level progress towards achieving the Sustainable Development Goals. On average, as you can see here, countries had reported 1 or more data points on about half of the indicators, and no country had actually reported data on more than 90% of the SDG indicators. This is a problem for high-income countries, but especially a problem for middle and lower-income countries as well. So on my final point I'd like to make is just about the importance of focusing on the local context as well. And this isn't a tension between the previous point. We can do global and we can do local, but we need to recognize that actually local actors are really key partners in achieving the SDGs. And what we need to do is support countries to ensure that they have the resources and the capacities to focus on the SDGs most relevant to their local contexts and their major challenges. And if they don't do this, there's a risk of a further misalignment between research priorities and the SDGs. And this, in turn, may then reduce the effectiveness of investments in research to address those goals. In Argentina, as you can see here, for example, there's many major challenges that exist in relation to SDG number 9, 10, and 15. But despite this, a lot of the research is actually focusing on priorities away from actually the kind of key SDG priorities within the countries. Similarly, within higher income countries, which often have the most unsustainable consumption patterns, generate more CO2 emissions, and contribute the most to climate change, they often do not specialize in research on the major environmental challenges relating to responsible consumption, climate action, or SDG 15, life on land. So there's this kind of disconnect that's really important to tackle. And this is a really important and growing area, as the focus of scientific research is also important in building local problem-solving capacities. And these capacities involve tacit knowledge building and sharing as well. And this also, in turn, highlights the value of building relevant local research capacity, including in informing local policy decisions through an understanding of context and the different pathways available to solve local and locally relevant research challenges. Often, a lot of our work at UNDP will be working with governments on STI pathways, and often many of these will be dominated by a handful of interests, a handful of political groups. So how do we make this conversation truly global, truly open, and make sure all people can be participants in defining what the local research priorities are? And this focus is also actually extends much more broadly. So we need to be building research and problem-solving capabilities, but also, where possible, doing knowledge transfer, capacity building, and technology transfer as well. So actually, this local focus is really important in identifying the most useful and relevant approaches and solutions. And one example I'm going to focus on here is about urbanization, as we're in Singapore. We do a lot of work on urbanization and cities at UNDP, and the smart city concept is one that's got a lot of interest globally. And often when we talk about sustainable development goals and urbanization, we focus on SDG number 11, so sustainable cities and communities. But what we want to do is actually, if we make smart cities and cities relevant to all locations, we make them truly smart cities, then they should be drivers of every single sustainable development goal. From tackling poverty and inequality, so SDGs number 1 and 10, through to the potential of precision agriculture for urban agriculture, and even supporting the biodiversity of cities in something like SDG number 15, life on land. But we can only pursue this range of different goals if we broaden what we mean by smart cities. So for example, if you Google smart cities, you get page after page of high-tech cities, laser beams firing all over the place, Internet of Things, 5G, often no people in these photos, no animals, no lives, no livelihoods. So a lot of our work at UNDP is about broadening and making the smart city concept more relevant to all settings, and broadening our definition beyond just the high-tech. So smart cities, for example, should be about how we use technology, whether high-tech, low-tech, or no-tech, and also innovation more broadly, to make the urban environment more inclusive, more people-centered, more livable, and more sustainable, fundamentally making cities about people. So if we broaden this definition, it then starts broadening the kind of solutions that we can use in the context of the SDGs. For example, if we broaden our definition of a smart city, we then look at Singapore, a high-tech city using high-tech solutions as an example of a smart city. We look at Curitiba in Brazil, which is home to one of the world's first bus rapid transit systems. We look at Harare in Zimbabwe. The picture you can see on the screen here is actually the Eastgate Shopping Center, which uses passive cooling to make the building environment comfortable, whether it's day or whether it's night. This saves significant money, significant energy, particularly as the country can experience very significant, considerable daily shifts in temperature. And actually, excitingly, this building was designed in 1996 by a local architect. Or we can look at somewhere like Kathmandu with its network of electric taxis, all many driven by female entrepreneurs, which have been in operation for more than 20 years. And again, this is another example of what we could call a smart city. And then coming full circle back to Singapore, again, using urban and nature-based solutions to soak up and managing rainflow. So when we're working with technology and science, we need to broaden the discussion and the discourse so we can find the solutions most relevant for the local context and the local needs and the local realities. And I'd just like to wrap up by mentioning 3 broader takeaways. The first is the importance of being open. So open to learning, open to new opportunities for synergies, as I mentioned, and open to changing our direction as priorities shift. And this also extends to openness in science. Our friends at UNESCO recently published their recommendation on open science, and they took a whole value chain approach here, from looking at open scientific knowledge to building open science infrastructure, and then also ensuring an open engagement and open dialogue within and beyond the research community. Secondly, we need to communicate, and science communication is a really important and enormously important and growing area, and actually something we'll be discussing in another event later this week with SG Innovate. We need to be sharing learning, sharing what does and doesn't work, sharing our failures most importantly, and also building feedback loops to improve what we do and what we've learned. And then finally, science is really important in informing decisions and decision making. I mentioned this earlier, but it's about ensuring that science, where possible, is about aligning with priorities and decisions, but it's also about building capacities in the public sector to leverage and understand science, insights, and data. And actually, a lot of our work is about working with the public sector to build capacity in the civil service, to leverage data, science, and other insights, to make policies and to make decisions. And then finally, and sadly, ending on a slightly more negative note, we also need to all benefit from science and share the gains of science wherever possible. As we continue to see with the global COVID-19 vaccine rollout, this doesn't always happen. Vaccines that are being developed through global scientific research, funding, and collaboration are not being globally distributed. And if the solutions generated by science are not equal, then nothing else will be equal either. We need to be thinking globally, locally, and inclusively wherever possible. So thank you all for your time. I really appreciate the platform and the opportunity. I'll share my email address as well. We're always very keen to learn from you all on the research side. We have, as I mentioned, country offices in about 170 countries around the world. So when you do all go home and you want connections locally, drop me a line. I'd be very happy to make those connections as well. But thank you.
Thank you, Mr. Henforth. We will now have a Q&A session moderated by Prof. Kishore.
Well, good evening, and thank you, Queloz, for that really wonderful overview of the role of science. And my take home is you identified 3 ways that science matters. One is understanding the indicators for the SDGs and just doing basic research to understand better where we're at and how we can therefore need to improve where the gaps are. And then you nicely talked about the science and technology knowledge that can produce innovative changes, including smarter cities. And then you also mentioned the institutional policy and governance challenges, and that was kind of your last take-home point. So I wouldn't mind beginning this moderation by asking you, how do you see the role of science in terms of those 3 areas? And the context to my question is, if you take the climate crisis, for example, which is, of course, one of the important objectives under the SDGs, the science, the indicators, is really strong. Yes. We have an international panel on climate change, 4,000 scientists, really strong. We even have technological innovations, solar panels, windmills, and so on, right? But everybody agrees the policy and governance lags. So what are your thoughts on how science can be part of closing that gap between the policy and governance we have today and what we need to achieve given the first 2 scientific findings?
This is a very big question. I think the first thing that science can be doing is, where possible, communicating, sharing research, sharing your insights. I spent quite a bit of time in government back in the UK, and often we were very, very close to having these kind of conversations. So keep shouting, keep yelling, keep sharing your work wherever possible to start closing that gap. The second, I think, is more on the kind of government side, is how do we start actually building science, technology, and innovation pathways that are actually more diverse. So bring in more voices into the discussion, bring in more solutions into the discussion, and actually making it more of a kind of multifaceted piece. These challenges are truly complex. They are what we call wicked challenges. There is no single solution to any of them. So we need a true kind of complex range of different solutions, actors, and individuals in that discussion. So where possible, providing platforms for that as well.
Thank you for that, and I appreciate both your practical knowledge and then your insights in your current position. I want to ask you a second question in the context of that, and one of the questions many social scientists work on is the question of policy durability, because we also see on the books really important policies that seem to reflect the science of what's needed and very important goals, but a history of, as time marches on, the goals get pushed off into the future. In fact, the SDGs replace the MDGs, replace the Rio Earth Summit's Agenda 21. So what are your thoughts on then how we might think about the durability question in the context of the diversity of stakeholders involved in these choices?
I think it's a really good point. So the Millennium Development Goals, which were a precursor to the SDGs, were more focused on low-income countries. So we already had a step forward with the SDGs, where they're truly kind of global conversations. So they're as relevant to a high-income country as they are to a low-income country. So already in durability, we're making this a kind of globally relevant conversation. But I think as well, where possible, it's about challenging to make sure that we keep the momentum going. What I've seen in government and working with governments is one of the most powerful forces is inertia. So you will run, and then things will kind of recalibrate back to the center or back to the status quo. So again, pushing and maintaining that momentum is very difficult. It's exhausting. And the challenges that we're all working on, we have been yelling about for a long, long time. And often we're not getting the kind of traction that we're looking to. So I think continuing that discussion is really important. What I see as really exciting is also just bringing in more partners into this conversation. So the private sector is a really interesting one that we work with a lot at UNDP, who often own a whole new part of the research agenda, who often have a different lens and a different voice to government as well. So bringing these kind of different actors into the conversation, I think, is really important in kind of maintaining that momentum and that durability as well.
I'm going to ask you one final question, and then I'm going to open it up to the audience and to the questions in Zoom land as well. But my third related question is about the different kinds of research knowledge needed for the indicators and the scientific indicators, the science and technology innovations, and the policy and governance challenges. And the background I'm a political scientist. I work on policy and governance. And 25 years ago, an article was written. And this is for you non-social scientists, what I call the real scientists. You guys that do the actual scientific work. Is that there was an article called "God Gave Physics the Easy Problems: Adapting Social Science to an Unpredictable World." Okay? And the idea there was, gosh, the social world, your item 3, is so confusing and changing. Norms change, ideas change, that you can't develop the same kind of Popperian hypotheses that you must adopt in your physical and biological sciences. So how do you guys handle that? You need different kinds of science when it comes to the third policy and governance challenge.
It's a really good point, and as a social scientist as well, we've done a lot of work around this. I think the most important point is, regardless of natural science, social science, any kind of research, is having that kind of feedback loop and having that dialogue. When we're doing work around policy, around research, or even the kind of interventions that we run, for us it's really important to have that dialogue with local communities, local partners, to make sure that firstly we understand actually what is and isn't working, but making sure that this is, we're feeding in what is the kind of relevant local context as well. So for example, we've been running a program quite recently to provide a platform for young people to involve themselves in digital transformation in a number of small island countries. And as part of that, it's about building the opportunities for them to actually engage in that dialogue as we're running these kind of programs. We can't just run forward and just do this in one go without talking to anyone anymore. It used to be a case that you would run a project, you would then do the monitoring and evaluation 2, 3, 5 years down the line. What we're learning now is actually this needs to be an iterative and an agile process. We need to course correct, we need to look at what isn't working, and actually make this a truly kind of transdisciplinary endeavor as well. So learn from the social sciences about measurement, learn from what's working on the natural science side as well, and also make this a truly kind of combined conversation.
Thank you. I'm now going to turn to some questions from Zoom land, and those of you in the audience, please feel free to line up at the mics. So there's 2 questions I'm going to ask together. They're around the pandemic, which, along with climate change, my colleagues and myself refer to as a super wicked problem. And the 2 questions are, one, where's the scientific progress helped when it came to, and where was that lacking when it came to tackling the corona pandemic? That's the first question. And the second one related is, what do you think is the biggest barrier to vaccine distribution in the low-income countries you mentioned?
So I think for the first one, I think what was one of the biggest benefits from around science is just how science worked for the past few years so effectively. Like delivering at pace, we saw research happening in real time, publications getting to journals in weeks instead of years, and actually a truly kind of global discussion and a truly kind of targeted discussion as well. So that was actually very interesting and very exciting to see that happen. The flip side to that, I think, is just how quickly research priorities shift. We had so much funding and money go into COVID-19 as it should have done. Sadly, that money was taken from other priorities, and now sadly we're seeing that actually money is being taken away from pandemic preparedness and away from healthcare as well. So it's actually how do we maintain this momentum, this durability, as you mentioned, in the context of funding, our research architectures, the institutional architecture as well, I think is really important. In terms of vaccine equity, this is a truly kind of multi-level question. There's things about IP, like how do we get this stuff into the countries that need it most? There are issues around distribution. How do we build and support healthcare systems to make sure vaccines get to the very last mile? Right. And then there are issues around politics and economics. How do we make sure that everyone has a voice at the table to make sure that they're getting the vaccines that they need? And it's a huge, huge conversation.
Thank you. Now, we have 3 people at the mics, and we only have about 5 seconds. So I'm going to ask, no, we have 3 minutes and 37 seconds, actually. I can see it right here in front of me. We're good to go. But I'm going to ask you to be pithy, and I'm going to ask all 3 of you to first ask your questions and then turn it over to Caleb afterwards. So over here on my left first.
Cool. Hello. My question is about some of these graphs you showed about the disconnect between the research that's actually being done and then the research that needs to be done for sustainable development. And it was just a question about how you think we need to tackle this discrepancy. At what level do we need to make changes to ensure that this gap gets smaller?
Okay, research gap question, thank you. Over here.
In magnetic resonance imaging, we've seen a great change in how research is done, but also what it leads to when the shift went from collaborating with, say, sub-Saharan African countries and having sub-Saharan African countries perform the research themselves on MRI. So I was wondering if you could comment on that in the context of these
great one. Internally building your capacity to generate knowledge. Great question. And the third one?
We just mentioned in the presentation slides that smart cities could solve most of the SDG problems and will obtain the goals. However, my question is, smart cities revolves around data, and how this data is being sent is using 5G or 5G advanced base stations or towers, right? And this actually will produce and increase carbon footprint and carbon emission. So I want to ask, is the benefit that comes from smart city will override the increase of carbon footprint that we will get? Thank you.
So a third question on not just synergies, but what we call in our lab whack-a-mole effects, things that actually get worse by doing things. Yeah, great questions, thank you.
So on the research side, this is a kind of historical issue where a lot of the research priorities in countries have been driven by decisions made decades ago by how institutions have been set up and funded, often by higher income countries. So there's a real kind of entrenched bias in this as well. Fundamentally, there's going to be a kind of redesigning of some of this global architecture, like how do we fund things better? How do we build local capacity? How do we do better technology transfer? How do we make sure that countries actually have the space and the resources and the capacities to actually focus on priorities most relevant to them? It won't happen overnight. I'll share actually a really good paper on this that I think is really relevant and that unpacks it in more detail, but it's a kind of historical issue. It's a big challenge.
Thank you.
On the, sorry, remind me, the MRI piece in terms of, yes, the Africa and how we, the kind of learning from there. The issue that we have here is that we do need to make this a kind of really collaborative and equitable piece. So providing opportunities for low-income researchers to be lead on publications, to do the research, to not keep the higher income elements. So in higher income countries, so often we see in many of the projects that we work in, you'll hire a data collector in a low-income country to do the work. The kind of lowest value-add component, where the higher income, the more value-add pieces stay in the higher income world. How do we rebalance that? But also fundamentally, it's how do we make these platforms available? From a lot of researchers I've worked with in lower-income countries, they can't even access conferences because they can't get visas. So how do we actually make these platforms more available to people, more accessible? Do we start putting platforms into these countries, which I think we should, and not just getting everyone to go to London, DC, New York, or elsewhere? So things like this, I think, are really important, getting you all in one place and lowering those barriers as well. On the smart cities point, apologies, my point wasn't that smart cities can do every SDG. It's more that often when we talk about smart cities, we focus just on SDG number 11. But if we actually broaden the definition of a smart city, it's relevant to all 17 SDGs. Incidentally, I hate the term smart cities. It's very tech-centric. It is about 5G, AI, big data. For me, a smart city is actually about technology and innovation. So high tech like 5G, big data, AI, but also low tech like frugal innovation. We do a lot of work around 3D printing, for example, and also no tech like nature-based solutions. So for me, a truly smart city is not 5G and AI. It's all these multifaceted solutions, and essentially it's about making a city more livable and environmentally sustainable and inclusive as well. So 5G is a good example. It can be a tool, but it's one tool in our kind of smart city toolkit.
So I want to jump in here now because we have these red zeros in front of us, which implies that the time's over. But I do want to just reflect two questions that we can't get to from Zoom land that I think are important to leave us with. And it's so impressive that what you've said in half an hour alone, so we're very excited for this beginning conversation. So one comment is about the openness question, which is that in many societies, for many good reasons, in fact, in all societies, we have policies that close information. That could be for IP rights, data restriction access rights, and so on. So how do we actually think then about openness when also sometimes you must have closed systems as well? And you can't answer these questions, I'm just raising them, sadly. Over dinner, you can engage them. And then secondly, there's a question from Christy Winkler, which is then, and I think is a really important one for all of you, how do we encourage individual scientists to feel responsible for ensuring that their research supports sustainable development? And that responsibility question, I think, is a tough one scientists have to grapple with because you're objective, but you're also citizens as well.
Completely.
And so let me just conclude by saying I think that Caleb Hanforth has done a nice job to show us how the UNDP, as an organization that brings science to action, in fact embraces both those imperatives. So thank you so much.
Thank you. Thank you, everyone.
Yeah.
Thank you, Mr. Hanforth and Prof. Kishore. We have now come to the end of the GYSS Day 1 sessions, and you may head over to the campus center where dinner will be ready at around 6:45 p.m. Thank you and have a good evening.
you.